Chapter 1. The Blueprint of Life. Understanding DNA. Imagine a vast library of genetic instructions, each word meticulously written in the language of life. This is DNA, the storybook of every living being. As you drift into this first chapter, you'll feel the calm unraveling of how DNA shapes who we are, like the hidden lines of a poem etched into every cell. We'll begin with the most fundamental building blocks of life, and look into how this script governs everything from eye color to the risk of diseases. DNA, or deoxyribonucleic acid, is like the book of life, a unique set of instructions found in every living organism. It is what makes you, you. It carries all the information needed to build and maintain an organism, from the tiniest bacterium to the largest mammal. DNA is present in almost every cell of your body, carefully packed inside the cell's nucleus. These instructions are not written in the way we normally think of writing, with letters and words on a page, but rather in a complex code made up of 4 molecules called nucleotides. These are adenine, a thymine T, cytosine C, and guanine G, often referred to by their initials. The arrangement of these 4 chemicals forms the genetic code that dictates everything about us. At first glance, DNA might seem complicated, like a jumble of letters, with no rhyme or reason. However, just like a book with a carefully crafted plot, there is a deep structure that allows DNA to store vast amounts of information. The DNA in every cell of your body is made up of 2 long strands, twisted together into a shape called a double helix. These strands are like the sides of a ladder, and the steps of the ladder are the pairs of nucleotides that bond together, always following a specific pattern. A pairs with T, and C pairs with G. This unique pairing system allows DNA to store and replicate information. This genetic code is what defines all living things. Think of it like a recipe or a set of blueprints. It tells your cells how to grow, how to function, and how to reproduce. It is the fundamental blueprint for building every part of you, from your eyes, skin, and hair to your organs, tissues, and the way your body fights off infections. The code inside your DNA directs every process in your body, controlling how you develop, how you heal, and even how your body responds to things like food, temperature, and stress. Each human being has about 20,000 to 25,000 genes, each gene being a segment of DNA that carries specific instructions. These genes determine things like your height, eye color, and even your ability to taste certain flavors. They also influence how your body handles disease and how it ages. Just like each chapter in a book brings a new piece of the story, every gene contributes a small part to the overall picture of who you are. Each gene has a role to play in the grand design of your body, and all of them work together in harmony to keep you alive and healthy. It's fascinating to think about how much of who you are is written into this code. Imagine that within your DNA is the history of your ancestors, stretching back for generations. The way your hair curls, the color of your eyes, and even your susceptibility to certain diseases are all influenced by the genes you inherited from your parents. As you reflect on this, it's humbling to realize that, in a sense, you carry the stories of generations that came before you. Their DNA flows through your veins, carrying their traits, their strengths, and even their weaknesses. In this way, your DNA is like a map, showing where you came from, who you are, and, in some ways, where you might be going. But what happens when something goes wrong in the genetic code? Imagine reading a book, only to find that one sentence is misspelled. It's a small mistake, but it changes the meaning of the entire story. In the same way, sometimes changes, or mutations, occur in the DNA that can affect how the body functions. These mutations can happen naturally, or they can be caused by things like environmental factors or lifestyle choices. While most mutations are harmless, some can cause diseases. For example, A mutation in a gene called the CFTR gene leads to cystic fibrosis, a genetic disease that affects the lungs and digestive system. These mutations can be passed down from one generation to the next, which is why some diseases run in families. If one of your parents has a mutation in their DNA, there's a chance that you could inherit it too. This is how many genetic diseases are inherited. Some conditions, like sickle cell anemia, are caused by a mutation in just one gene, while others, like certain types of cancer, are caused by changes in multiple genes. However, not all mutations are harmful. Some mutations can actually be beneficial. Over time, small changes in DNA have allowed life to adapt and evolve. For example, in some populations, a mutation that allows people to digest lactose, the sugar found in milk, became advantageous in areas where dairy farming was common. This mutation helped people survive in environments where other food sources were scarce. Mutations are part of the reason why life evolves over time, and they allow species to adapt to new challenges. Despite the occasional problem that a mutation can cause, the overall system of DNA is incredibly efficient and resilient. Your cells are constantly reading and copying your DNA, repairing any mistakes that may happen along the way. The cell has its own proofreading system to ensure that the instructions are followed correctly. When things go wrong, your body can sometimes fix them, but if the mutation is too severe or too widespread, it can cause health problems. It's amazing to think about how such a small set of molecules can carry so much information. Everything that makes you who you are, your personality, your body's responses, your vulnerabilities and strengths, is written into this genetic code. And yet, this code is not fixed. It is constantly being read, copied, and sometimes edited. Just like a book can be revised or rewritten, DNA can sometimes change over time. These changes can occur in small ways, like a typo in a sentence, or they can be more significant, like a chapter being added or removed. But even as DNA changes, it remains the foundation of life itself. As we think about the incredible complexity of DNA, we can begin to understand why scientists have been so fascinated by it. The study of genetics is like opening a treasure chest, full of mysteries, each discovery leading to another. With each new breakthrough, we uncover more about how life works and how we might be able to harness this knowledge to improve our health, solve problems, and even prevent diseases. The study of DNA has already led to many advances in medicine. By understanding how genes work, scientists can develop treatments for genetic diseases, like gene therapy. This therapy involves replacing or repairing faulty genes in order to treat or prevent disease. Imagine a world where doctors can correct the genetic mistakes that cause diseases before they even begin. The potential is vast, and we are only scratching the surface of what we can achieve. Now, as you begin to drift into a deeper sense of understanding, the idea of DNA and its role in our lives becomes even more intriguing. What happens when we can edit this blueprint, changing it to heal disease, or even improve the way we live? This thought leads us gently into the next chapter, where we explore the power of gene editing and how it's already beginning to change the way we approach medicine and life itself. The question lingers in the quiet space of your mind. What if we could not only read the story of life, but rewrite it? Chapter 2. The Evolution of Genetics. From Mendel to modern science, step into the world of a scientist's curiosity, where ancient pea plants led to modern breakthroughs. You'll gently trace the history of genetics, from the humble beginnings of Gregor Mendel's pea experiments to the revolutionary discoveries of today. Along the way, we'll reflect on how our understanding of life itself has evolved. Each discovery like a stepping stone leading us to deeper truths. In the quiet stillness of a garden, where delicate flowers sway in the breeze, an idea began to grow, an idea that would eventually change our understanding of life itself. This garden, though simple, was the birthplace of one of the most important discoveries in the history of science. It all began with Gregor Mendel, a monk who, like so many great thinkers, was driven by a deep curiosity about the world around him. Mendel's journey started with something very small: pea plants. These plants, with their delicate flowers and round, smooth seeds, seemed to hold a quiet secret. Mendel carefully crossed different varieties of these plants, recording their traits with patience and precision. His work, though simple in its methods, revealed a profound truth. Traits, like flower color or seed shape, were passed down from one generation to the next in a way that followed predictable patterns. It was as though life itself was following an unspoken set of instructions. Mendel discovered the basic rules of inheritance, rules that would eventually become the foundation of genetics. Imagine Mendel's excitement as he realized that the pea plants were following a hidden code that had been passed down through countless generations, undisturbed and constant. It was like discovering a secret language, one that explained how life itself was passed from one form to the next. Mendel's experiments with pea plants introduced the world to the concept of dominant and recessive traits, concepts that would become key to understanding the transmission of genetic information. He could explain why some traits, like purple flowers, were more common, while others, like white flowers, appeared only when both parent plants shared a specific trait. Though Mendel's work was groundbreaking, it wasn't fully understood in his time. His discoveries lay dormant for many years, hidden away like an undiscovered treasure, until other scientists began to build upon them. As time passed, the world began to realize that Mendel's pea plants were only the beginning. The field of genetics would grow, like a tree with roots that stretched deeper and branches that reached higher, uncovering secrets about life that were once beyond our grasp. The next step in this journey took us from the fields of pea plants to the microscope, where a new chapter in the story of genetics began. In the late 19th and early 20th centuries, scientists began to study cells in more detail. They discovered that within the nucleus of each cell was something incredible: chromosomes, the structures that carried the genetic information Mendel had hinted at. These chromosomes were like books, holding volumes of data that could be read and passed on to the next generation. As technology improved, the study of genetics grew more complex. The microscope revealed more about how traits were passed from one generation to the next, and scientists realized that chromosomes were the carriers of Mendel's genes. But even this discovery was only part of the story. Scientists began to suspect that there was more to the genetic code, something hidden within the chromosomes that controlled not just how traits were inherited, but how they were expressed. The answer came in 1953, when James Watson and Francis Crick, working with Rosalind Franklin's X-ray images, uncovered the structure of DNA. DNA was the key to understanding the instructions within the chromosomes. It was like the master blueprint, the key to unlocking the mysteries of life. DNA's double helix structure, with its twisting ladder of nucleotides, provided the framework for understanding how genetic information was stored and passed on. This was a revelation, a moment when everything began to fall into place. Imagine how the world felt when this discovery was made, the realization that inside each of us, in every cell, was the secret to life's code. DNA, with its 4 basic components, cytosine, thymine, cytosine, and guanine, could carry vast amounts of information in a simple, elegant form. It was like a puzzle that, once solved, revealed the intricate design of life itself. And with this understanding, the path to understanding diseases, evolution, and the very essence of what it means to be alive opened wide before us. From there, science began to accelerate. The sequencing of the human genome, completed in 2003, was a monumental step. Scientists mapped out all 20,000 to 25,000 genes in human DNA, creating a detailed picture of the genetic instructions that govern every part of our bodies. It was like reading a giant book that detailed the story of human life. But as much as we learned, the more we realized there was still so much more to uncover. Genes were not just simple instructions. They were part of a much more complex network of interactions that determined how we grow, how we heal, and how we age. This journey through the history of genetics shows us that science is not a straight line, but a winding path, with each discovery building upon the last. From Mendel's simple pea plants to the Human Genome Project, our understanding of genetics has evolved over centuries. And with every discovery, we've come closer to understanding the deep mysteries of life. But the story doesn't end here. There is still so much we don't know. Even today, scientists are unraveling the complexities of gene expression, epigenetics, and how our environment interacts with our genetic makeup. Every new discovery is like a page being turned in a book that has no final chapter. With each piece of the puzzle that is uncovered, we move closer to understanding not just who we are, but how we can shape our future. And as we pause for a moment, reflecting on all that we've learned, a new question emerges. How far can we go with this knowledge? If we can understand the code that makes us who we are, can we change it? What happens when we start to write our own chapters in the story of life? This thought lingers, gently guiding us into the next chapter, where we will explore the possibility of editing this code to heal diseases, enhance our abilities, and perhaps even design the future of life itself. As you contemplate the vastness of what we've learned, a quiet thought begins to take shape. Could we one day rewrite the story of life itself? Chapter 3. A single letter in a giant code. What are genes? Like the delicate threads in a spider's web, genes are the patterns that connect us to all life forms. This chapter invites you to contemplate the subtle elegance of genes, those tiny pieces of DNA that determine everything from our height to our hair color. With every whisper of the narrative, you'll come to understand how one letter in this genetic code can change everything. Genes are the quiet architects of life, the hidden threads that bind us together, and yet they are so small, so seemingly insignificant in the vastness of existence. Imagine them as tiny, intricate instructions tucked away within the great blueprint of your being. Each gene is a sequence of letters, just for in total, A, T, C, and G. Yet, from this simplicity, the complexity of life unfolds, like a delicate scroll revealing a story too vast to fully comprehend. These 4 letters, so simple and yet so profound, create the instructions that form who we are, from the color of our eyes to the height of our frame. They are like the notes of a song, where each note is a letter, and when arranged in the right order, they create a melody of life. The beauty of genes lies in their subtlety, for they are the quiet whispers of what we are, passed from one generation to the next, constantly shaping and reshaping the fabric of existence. Picture the elegance of a spider's web. Each thread connects, forming a delicate pattern, and if one thread is out of place, the entire web can shift and change. In the same way, genes are the threads of life. They form patterns that determine our appearance, our behaviors, and even how our bodies react to the world around us. Just as the spider weaves its web carefully, each gene is placed with precision, a part of a greater design. And yet, in this intricate tapestry, a single thread, a single letter in the code, can alter everything. A single change, a mutation, can shift the entire pattern, sometimes subtly, sometimes dramatically. Consider how one small letter, a single change in the genetic code, can make all the difference. Think of sickle cell anemia, a condition caused by just one letter being altered in the sequence of the gene that codes for hemoglobin. A single change that leads to the bending of red blood cells into a crescent shape instead of their normal round form. It is a change so small, so seemingly insignificant, yet it has profound effects on the body. This is the delicate power of genes. They hold within them the potential to shape life in ways that are both subtle and immense. But not all changes are harmful. In fact, some of the most beautiful variations in life come from changes in our genes. The vast diversity of human beings, the different colors of skin, the shapes of faces, the heights of bodies are all due to variations in the genetic code. These variations are not mistakes, but rather the creative dance of nature, continually finding new ways to express the possibilities within life. Just as a painter mixes colors to create new hues, so too does the genetic code mix and match its letters to produce the endless variety of life forms we see in the world. This intricate dance of genetic variation is not limited to humans. It extends to every living being. In the animal kingdom, in the plant world, in the unseen world of microorganisms, genes are the guiding hands that shape life. The petals of a flower, the wings of a bird, the cells that make up the muscles of a whale, every detail, every feature, is encoded within genes. The butterfly's wings, for instance, with their delicate patterns and vibrant colors, are not the result of random chance, but the outcome of millions of years of genetic instruction. The genes of a butterfly guide the formation of its wings, shaping them with the precision of an artist. Yet, as vast and intricate as genes are, they are not static. They are constantly in motion, changing and adapting, responding to the environment, to the challenges life faces. In this way, genes are like a river, always flowing, constantly evolving. Sometimes, the changes happen quickly, creating new traits, new forms of life. Other times, the changes are slow, like the gradual shaping of a landscape over centuries. But regardless of the pace, the dance of genes never stops. What is perhaps most remarkable about genes is their ability to carry the history of life itself. Within each gene lies a record of the past, a story of survival, of adaptation, of change. As you trace your lineage, from your parents to your grandparents, and beyond, you carry with you the story of countless generations. Your genes are the echoes of your ancestors, their traits and experiences woven into the very fabric of your being. And as you pass on your genes to your children, you continue the cycle, sharing the story of life in its endless flow. But genes are not just about the past. They are also the keys to the future. The genetic code is the blueprint that guides evolution, allowing life to adapt and survive in an ever-changing world. Through the slow, steady accumulation of small changes, life evolves, finding new ways to thrive in different environments. The variety of life we see around us, from the tallest trees to the tiniest insects, is the result of this continuous process of genetic change. And as we move forward, our genes continue to evolve, shaping not only who we are today, but who we will become in the future. This understanding of genes opens up a world of possibilities. If we can understand the code that shapes life, can we not also learn to read it more clearly? Can we, perhaps, learn to rewrite parts of it? What would it mean if we could edit genes to cure diseases, to enhance human abilities, or even to prevent the genetic conditions that have shaped our history? Could we, as humans, become the authors of our own genetic story, editing and rewriting the chapters of life itself? These questions, though fascinating, are not without their complexities. Editing genes is not as simple as changing a word in a sentence. The genetic code is a vast, interconnected web, and altering one part can have far-reaching consequences. A change in one gene might affect others in unexpected ways, leading to outcomes we cannot yet predict. It is a delicate balance, like a musician tuning an instrument, where each adjustment must be made with care. But the potential of gene editing is undeniable. It holds the promise of a future where we can cure genetic diseases, where we can enhance the human body's natural abilities, where we can even design the life we want to live. As we explore this possibility, we are at the threshold of a new chapter in the story of Leaf, a chapter where we begin to write not just the words of our own existence, but perhaps the words of the future. And so, as we reflect on the power of genes, the quiet architects of life, we are left with a lingering thought. What if we could edit this code, reshaping the very foundations of life itself? What changes could we make, and what would those changes mean for the world we live in? As you rest in the calm of this reflection, the thought arises: how far can we go with the power to rewrite the genetic code? Chapter 4. Mutations. When the code goes awry, in the quiet stillness of the night, consider the slight imperfections that emerge in nature. Mutations are like small ripples in a pond, some harmless, others life-changing. We'll explore how mutations in the genetic code can sometimes bring about diseases, and how life has adapted to such changes over millions of years. As you drift deeper into the rhythm of your breath, imagine, for a moment, a tiny ripple spreading across the surface of a calm pond. It begins so small, so insignificant, yet it has the power to disturb the stillness and create waves that travel outward. These ripples are like mutations, tiny changes in the genetic code that, though small at first, can sometimes lead to profound effects. Mutations happen all the time, quietly and without notice, deep inside our cells. They are like little glitches in a perfectly written story. A single letter misplaced in a word, or a slight shift in a carefully drawn line. Most of the time, these changes go unnoticed, like a breeze that barely rustles the leaves. They might not change the overall picture, and life continues on its path, uninterrupted. But sometimes, these small alterations in the genetic code can ripple outwards, creating changes that are far more noticeable. They can lead to new traits, or, in some cases, diseases. To understand mutations, imagine the genetic code as a long string of carefully arranged beads. Each bead is a letter, one of the 4 building blocks that make up our DNA: adenine, thymine, cytosine, and guanine. These letters form the words and sentences that guide our development and function. Now, picture that one bead, just one, shifting out of place. This small change, this slight distortion in the sequence, can have consequences far greater than we might expect, just as a single misplaced bead in a necklace can throw off the entire pattern. A mutation in a gene can change how the body functions. Most mutations, however, are not harmful. They are simply variations, like a unique mark on a rock or a different shade of green in a forest. These changes don't disrupt the flow of life. Instead, they add to the rich tapestry of diversity that defines all living things. Just as no 2 leaves are exactly the same, no 2 people have the exact same genetic makeup. These mutations may influence things like eye color, height, or even the way our body reacts to the environment. Over time, such changes can accumulate, leading to the beautiful diversity we see in the world around us. But there are times when a mutation does disrupt the balance of life. Just as a single stone thrown into a pond can cause waves that change the surface of the water, some mutations can create ripples within the body that lead to disease. These mutations can change how the body works, sometimes in ways that make it harder for the body to function as it should. Think of cystic fibrosis, a condition caused by a mutation in a gene responsible for producing a protein that helps regulate salt in the body. This mutation leads to the buildup of thick mucus in the lungs and other organs, causing difficulty breathing and digesting food. The remarkable thing about mutations is that, like the ripples in a pond, they are not always predictable. Some mutations lead to diseases that can be passed down from one generation to the next, while others may arise unexpectedly. Some mutations, like the one responsible for sickle cell anemia, can even provide an unexpected advantage. In regions where malaria is common, People with sickle cell trait are less likely to develop severe forms of malaria. In this way, a mutation that might normally be harmful becomes a shield, protecting individuals from a deadly disease. Mutations are the quiet architects of evolution, the unseen hands that have shaped life over millions of years. As mutations accumulate and spread through populations, they provide the raw material for natural selection. This process, slow and steady, allows life to adapt to its environment, responding to challenges and finding new ways to thrive. In this way, mutations are the sparks that ignite the fires of evolution, allowing life to adapt to new conditions, survive in different environments, and evolve into something new. But while mutations are a driving force behind evolution, they are not without their consequences. Sometimes, the changes they bring are too disruptive, too powerful for life to adapt to. In these cases, the mutation can lead to diseases, conditions that make survival more difficult. Yet, even in the face of such challenges, life finds ways to adapt. The body's ability to heal, repair, and sometimes compensate for genetic changes is a testament to the resilience of life. Even when things go awry, life seeks balance, finding new ways to move forward. There is something inherently beautiful in the way mutations are part of the ever-evolving story of life. Though they can bring challenges, they also bring possibilities. Just as a storm might shake the branches of a tree, but in doing so, create space for new growth, so too do mutations provide opportunities for life to evolve and grow in unexpected ways. Life in its wisdom adapts and changes, finding new paths when the old ones no longer serve. As we consider the role of mutations in the story of life, we begin to see how the tiny changes that seem insignificant can have such powerful effects. A single letter in the genetic code, a small shift, can lead to the creation of entirely new traits, or the emergence of diseases. These changes remind us of the delicate balance that life depends upon, and the constant dance between change and stability. In the quiet of this reflection, you might begin to wonder, what happens when the genetic code itself can be rewritten, when the ripples caused by mutations are not just natural occurrences, but intentional changes made by human hands? What could we achieve if we could shape the genetic code as easily as we shape words on a page? The next chapter calls us to explore this possibility, where the science of gene editing holds the promise of rewriting the very instructions of life. And so, as you reflect on the small, subtle changes that shape the world, a thought lingers in the calm silence. What if we could intentionally edit the genetic code to prevent mutations, or even create new possibilities? Chapter 5. The Silent, Suffering Genetic Diseases, and Their Toll. As we drift into understanding the impact of genetic diseases, think of them like silent storms inside the body. These invisible forces can alter a person's life in profound ways. We'll gently explore a few well-known genetic conditions, reflecting on the struggles they bring, and the silent strength of those who endure them. Imagine the calmness of a peaceful landscape, the stillness of the morning, when suddenly, unseen forces begin to shift. A storm brews quietly, gathering strength beneath the surface, though no one can see its arrival. It is not a storm you can hear or see, yet its effects are profound. These invisible forces are much like genetic diseases. They lurk within the very fabric of our being, silently altering lives, sometimes in ways that are hardly noticeable at first, but with consequences that echo through the days, months, and years. Genetic diseases are like the quiet storms inside the body, born from small changes or disruptions in the genetic code. They are often inherited, passed from one generation to the next, like whispers in the wind that carry from the past. These diseases can range from mild to severe, and though their effects are not always visible on the surface, they quietly influence the way a person lives, moves, and experiences the world. In many ways, These diseases are silent, hidden from view, yet they shape the lives of those who carry them, their families, and their communities. Take, for example, cystic fibrosis, a genetic disease that affects the lungs and digestive system. This condition arises when a mutation occurs in a gene responsible for regulating salt in the body. Imagine your body's internal environment as a delicate balance, like a perfectly balanced ecosystem. The body needs this balance to maintain its rhythm, its flow, but in cystic fibrosis, the balance is disrupted. The lungs fill with thick, sticky mucus, making it difficult to breathe. The digestive system struggles to process food, and daily activities that most of us take for granted become a challenge. What is perhaps most striking about cystic fibrosis and other genetic diseases is how quietly they unfold. It's not always something visible, something that others can see or touch. It's a storm that rages within, a silent force that shapes a person's life from the inside out. And yet, those who live with genetic diseases carry a resilience that is often unseen. They face challenges with a quiet strength, enduring the difficulties that others may not fully understand. Sickle cell anemia is another example, a genetic condition that affects the shape of red blood cells. In those with sickle cell anemia, the red blood cells, which are normally round and flexible, become crescent-shaped and rigid. These sickle-shaped cells can get stuck in small blood vessels, causing pain and limiting the flow of oxygen to various parts of the body. The pain can be excruciating, yet it is not always visible to the outside world. From the outside, the person looks just like anyone else, but inside, the body is struggling, fighting to keep the blood flowing, to maintain life's quiet rhythm. This invisible suffering, the pain of a disease that cannot be seen but is felt deeply, is often accompanied by a silent strength. Those who live with genetic conditions often learn to navigate life in ways that others cannot fully comprehend. Their resilience is not only in their ability to endure the physical challenges, but also in their emotional strength, their determination to live fully, despite the limitations that their conditions may impose. It is important to remember, though, that not all genetic diseases cause suffering in the same way. Some conditions are mild, with little impact on a person's daily life. Others, like Huntington's disease, gradually take away the abilities that were once so familiar. Huntington's disease is caused by a mutation in a gene that leads to the gradual breakdown of nerve cells in the brain. The symptoms begin slowly, but over time, they interfere with movement, memory, and emotional control. For those who inherit this condition, the knowledge that it will eventually take away so much is a silent burden to bear. And then there are those who live with genetic diseases that don't always manifest right away. Diseases that hide in the body for years, only to reveal their presence when it is perhaps too late to do much. These conditions often cast a shadow over a person's life, where they are constantly waiting for the storm to break, for the invisible forces to make themselves known. Yet, even in this uncertainty, there is strength in the human spirit. There is a quiet courage in those who live with the knowledge that their bodies carry the potential for change, for illness, for difficulty, and yet they continue to live, to love, and to dream. The struggle with genetic diseases is not just about managing physical symptoms. It is about learning to live with the unknown, accepting that some parts of life are beyond control. It is about finding peace within the unpredictability, learning to navigate life's challenges with grace. It is about strength that cannot be measured, a resilience that lies in the spaces between the struggles. In the quiet moments when the body is still, through the suffering that genetic diseases bring, we also see the beauty of human connection. Families and communities come together to support those who are affected. There is love and compassion that rise out of the silent suffering, forming bonds that transcend the limitations of the body. It is in these moments, in the small gestures of care and understanding, that we find the true strength of the human spirit, As we reflect on the silent storms of genetic diseases, we begin to understand the deep complexity of life. These diseases are not simply obstacles. They are a part of the intricate pattern that makes us who we are. They remind us of the fragility of life, and yet, they also remind us of the incredible strength we possess to endure, to adapt, and to continue. And, as we begin to understand the impact of these hidden forces, we are left with a question. What if there was a way to rewrite the code that causes such suffering? Could we, through science and technology, alleviate the burden of genetic diseases and change the course of someone's life? This thought brings us gently to the next chapter, where we explore the possibilities of gene editing and the power to reshape the future. As you rest, the question lingers softly in your mind. Could we one day rewrite the genetic code to heal the suffering that silent storms bring? Chapter 6. The Promise of CRISPR. A glimpse into the future. Now imagine a tool that could gently rewrite the book of life, fixing imperfections in the genetic code as a writer might edit a manuscript. This is the promise of CRISPR, a technology so precise that it can alter genes with the delicacy of a sculptor refining a marble statue. We'll explore how CRISPR is changing the way we think about genetic diseases. Picture, for a moment, the elegance of a finely crafted manuscript. Each word is perfectly chosen. Each sentence flows with purpose. But then, imagine that one word is out of place. One sentence doesn't quite fit the rhythm of the story. Now, think of a tool, delicate and precise, that can gently erase that one imperfection and replace it with the right word, effortlessly restoring the beauty of the entire text. This is what CRISPR, a powerful tool in genetics, promises: the ability to rewrite the story of life. Editing the genetic code with the same care a writer takes to perfect a manuscript, CRISPR is like a pair of molecular scissors, capable of cutting and pasting the genetic instructions that make us who we are. These scissors are not random, but incredibly precise, able to target and edit specific parts of the DNA sequence with remarkable accuracy. Just as a sculptor shapes marble, with gentle strokes, CRISPR can refine the genetic code, correcting errors that cause diseases, or unlocking new possibilities for human health. It is as though life itself is being rewritten, one letter at a time, and with it, the potential to change the course of many lives. Imagine the immense power of such a tool, the ability to go into the very blueprint of life, to identify the tiny flaws that lead to genetic diseases, and to repair them before they can cause harm. For centuries, humanity has struggled with diseases that have been passed down through generations, inherited silently, often with no way to stop them. But with CRISPR, we stand at the edge of a new era in medicine, where these inherited flaws could be corrected, where genetic diseases, like cystic fibrosis, sickle cell anemia, and even some forms of cancer might one day be healed. The idea of CRISPR was born from a natural process. In bacteria, CRISPR functions as an immune system, a way for the organism to protect itself from viruses. When a virus attacks, the bacteria remembers the invader's genetic code and stores it in the form of CRISPR sequences. These sequences act like a genetic memory, allowing the bacteria to cut out and destroy the virus the next time it invades. Scientists, in their curiosity and ingenuity, realized that this process could be harnessed to edit the DNA of more complex organisms, including humans. This discovery opened up a world of possibilities. With CRISPR, scientists could now target specific genes and make changes with precision. Imagine being able to edit the gene that causes a hereditary condition, cutting it out like a typo in a sentence. Or envision a future where a child is born without the risk of inheriting a debilitating disease, simply because the gene responsible for that condition was edited before birth. This technology has the potential to alter the very course of genetic inheritance, offering hope to families who have lived with genetic diseases for generations. One of the most promising aspects of CRISPR is its ability to make changes at the very start of life. In the past, genetic diseases could only be managed after they had already caused harm. But with CRISPR, there is the possibility of preventing the disease from taking root in the first place. It's as if we could go back to the beginning of a story and rewrite the opening lines, ensuring that the narrative unfolds without the disruption of disease. The potential here is vast, offering a chance for a future where many genetic conditions are not just managed, but completely eradicated. But as with any powerful tool, CRISPR comes with its own set of challenges and ethical questions. While the potential to cure genetic diseases is extraordinary, there is also a responsibility to wield this power with care. What happens when we begin editing the genetic code of not just the individual, but of entire generations? What are the long-term effects of altering the human genome? These are questions that scientists and ethicists are still grappling with, as they work to ensure that CRISPR is used responsibly and safely. In the midst of this uncertainty, one thing remains clear. CRISPR is already changing the way we think about genetics. It is challenging our understanding of what is possible and pushing the boundaries of medical science. Just as the invention of the microscope allowed scientists to see the hidden world of cells and bacteria, CRISPR is allowing us to see and change the hidden world of our genetic code. It is as though we have discovered a new way to read and write the book of life, and with that knowledge comes the opportunity to rewrite the chapters that have been marred by disease. As we stand at this crossroads, we are reminded of the delicate balance between power and responsibility. The ability to edit genes, to rewrite the very code of life, brings with it both incredible promise and profound questions. How far should we go in altering the human genome? What boundaries should we set, and what consequences might arise from these changes? In this moment of reflection, you might feel the quiet pull of curiosity. What comes next in this unfolding story? What other possibilities does CRISPR hold? Will we one day be able to edit not only genetic diseases, but also enhance human abilities, increase resilience, or extend life? As we explore these questions, we are reminded of the potential that lies ahead, and of the gentle promise that CRISPR offers, a glimpse into a future where the book of life is no longer bound by the limits of genetic disease. As the echoes of these possibilities begin to settle in your mind, a soft thought lingers. What happens when we take the next step and begin to rewrite not just disease, but the very essence of human potential? This thought leaves you wondering, how far will the story of gene editing take us, and what boundaries will we set along the way? Chapter 7. How CRISPR works. Cutting and pasting life's code. Envision a pair of molecular scissors, delicately cutting and pasting the letters of life's great story. This is CRISPR at work. In this chapter, we'll take a slow, meditative journey through the steps of CRISPR's mechanism, understanding how it can remove faulty genes and replace them with healthy ones. Imagine a pair of delicate, invisible scissors floating through the depths of your body. These scissors are not like any scissors you've used before, for they are capable of cutting and pasting the very fabric of life itself. They work with a precision that seems almost magical, capable of cutting through DNA, the long, twisting strand that holds the code of life, and replacing the faulty parts with something new, something better. This is the essence of CRISPR, a technology that has brought the power of gene editing to the forefront of science. Just as a writer might edit a sentence to make a story clearer, CRISPR allows scientists to edit the story of life. It's a tool that can pinpoint the exact place in the genetic code where a change has gone wrong and then make a correction. It's as if, through the delicate hands of this molecular scissors, life itself is being rewritten, one letter at a time. The process is like a quiet, thoughtful revision, slow and precise, carefully selecting which pieces of the genetic code need to be removed and which need to be replaced. But how does CRISPR know where to make these edits? Picture for a moment a map. Every spot on that map represents a letter in the long string of DNA. CRISPR is guided by something called RNA, a molecule that helps carry the genetic instructions. RNA is like a messenger, bringing the precise coordinates of where to cut. It is this RNA that tells the molecular scissors exactly where to go, how to make the cut, and where to leave the new piece of genetic material. Now, imagine that RNA is a soft whisper guiding the scissors along the path. The scissors, through this guidance, arrive at a specific location in the DNA strand, just where the faulty gene resides. With a gentle snip, they cut the strand, and then the real magic happens. The gene can now be replaced, like a broken word, in a story being swapped for the correct one, or an old phrase being refreshed to bring new meaning. The body's natural repair systems then come in to complete the process, sealing the new piece of DNA into place, ensuring the story flows smoothly once again. What makes the CRISPR so powerful is its ability to make these changes with such precision. Imagine trying to fix a page in a large book without damaging the pages around it. It's no easy task, yet CRISPR achieves this with remarkable accuracy. This precision is what gives CRISPR its potential to treat genetic diseases. By targeting the very source of the disease, a faulty gene, and replacing it with a healthy version, CRISPR offers a way to correct the genetic mistakes that have been passed down through generations. Think about a genetic disease like cystic fibrosis, for example. This condition is caused by a mutation in a gene that affects the way the body handles salt, leading to thick, sticky mucus in the lungs. In the past, treating cystic fibrosis meant managing the symptoms, but with CRISPR, the possibility exists to go deeper, to correct the gene itself. The molecular scissors, guided by RNA, would find the faulty gene and replace it with a healthy one, allowing the body to function as it was intended to. The potential to heal in this way is a profound step forward, a way to rewrite the genetic code and give people a new chance at life. But CRISPR's powers extend beyond disease. This technology also holds the promise of advancing our understanding of biology in ways that were once unimaginable. It opens the door to experimenting with genes, to discovering new ways to improve health, enhance resilience, and even prevent diseases before they can even start. As we stand on the precipice of this new era, CRISPR offers a glimpse into a future where we can shape life with greater care, where we can repair the underlying causes of illness, and perhaps even prevent them altogether. Yet, as with any powerful tool, there is a need for caution. The ability to edit genes, to change the very fabric of life, brings with it a responsibility. There are ethical questions to consider. What happens if we begin to edit genes, not just to treat disease, but to enhance our abilities, to change physical traits, or even intelligence? Where do we draw the line between healing and enhancement? These are questions that will need to be answered as CRISPR moves forward, questions that require careful thought and consideration. In the quiet stillness of this moment, consider what it would mean to have the ability to edit the human genome with such precision. What if, in the future, we could prevent diseases before they could even begin to take hold? What if we could correct genetic errors that cause suffering, offering relief to those who have long been burdened by inherited conditions? The potential to reshape life in such a way is both exciting and humbling, for it asks us to consider the very essence of what it means to be human. CRISPR is not just a tool for today. It is a glimpse into tomorrow. It is a technology that could unlock new possibilities for medicine, for human health, and for the future of life on Earth. It reminds us that, just as a writer can edit a sentence, so too can we edit the story of life itself, making changes that can heal, that can enhance, and that can transform. Yet with this power comes the need for wisdom, for careful thought, and for an understanding of the consequences of our actions as we look toward the future. The questions that arise are as much about ethics and responsibility as they are about science and possibility. What do we do with this newfound ability to shape life? And how do we ensure that we use it in ways that truly benefit humanity? The answers to these questions will shape the direction of this new chapter in the story of life. As you reflect on the possibilities of gene editing, a quiet thought lingers. What will we do with this power to rewrite the genetic code, and how will it shape the future of life on Earth? Chapter 8. Editing Genes. What it means for us. Picture a world where we can correct genetic errors before they even begin. A future where we can stop genetic diseases before they start. This chapter takes you into the heart of gene editing potential, pondering what it could mean for us as individuals and as a society. Imagine, for a moment, a world where illness no longer has to follow the same old path. In this world, the stories of genetic diseases, inherited silently from one generation to the next, can be rewritten. Picture yourself standing at the threshold of a future where we can stop these diseases before they even begin. A place where the power to edit genes offers a new kind of hope. This is the promise that gene editing and tools like CRISPR bring into focus the possibility of a world where our genetic inheritance is no longer a burden but an opportunity for change. When we talk about editing genes, we are speaking of the ability to go into the very fabric of our biology, to find the strands that have been miswritten or damaged, and to gently correct them. Imagine it as a kind of restorative healing, not of the body itself, but of the instructions that govern the body. Genes are the blueprint of life, the set of instructions that guide every process within us, from the beating of our hearts to the colors of our eyes. If a part of this blueprint is flawed, it can lead to diseases and disorders that affect everything from our development to our health. But with gene editing, we have the power to correct those errors, to make changes that could potentially prevent these conditions from taking root in the first place. This idea is still so new, so transformative, that it's easy to get lost in the excitement of what could be. But it is also worth reflecting on what it would mean to change the very nature of our genetic code. Imagine the lives that could be saved, the suffering that could be avoided, and the families that could be spared from the pain of inherited conditions. Take, for example, the possibility of using gene editing to eliminate inherited diseases, like Huntington's disease, where the genetic error that causes the condition could be corrected before it ever manifests. In such a future, children born into families where genetic diseases once ran through the bloodlines could be free of that inheritance, untouched by the conditions that have burdened generations before them. But what happens when we have the ability to edit genes, to change the very essence of who we are? The question, though hopeful, also brings with it a sense of responsibility. What limits should we place on this technology? It is one thing to correct errors in a gene that leads to a disease, but what about enhancing the genetic code in other ways? What if, instead of just eliminating diseases, we could also enhance human traits? Could we increase intelligence, improve physical abilities, or even extend life itself? These questions bring us to the heart of the ethical dilemmas surrounding gene editing. The power to alter the genetic code is immense, and with it comes the responsibility to use it wisely. As you reflect on this, imagine what it would mean for you personally if the chance to correct genetic flaws were available. What if you could remove the risk of certain diseases that may have affected your family for generations, before they even had a chance to emerge? This would not just be a gift for individuals. It could reshape society itself by reducing the number of people born with genetic disorders. We could create a world where the burden of disease is greatly reduced, where the need for treatments and interventions is lessened, and where people could live their lives with fewer health concerns, all thanks to the ability to rewrite the genetic code. Yet, with such power comes a deeper question. What does it mean to alter life in this way? The ability to edit genes, to correct or enhance them, asks us to think about the very nature of what it means to be human. Is our humanity defined by the genetic traits we inherit? Or is it more about the experiences we have, the choices we make, and the lives we live? These questions tug at the fabric of our understanding, inviting us to ponder not just what we can do, but what we should do with this incredible power. As we explore these possibilities, we also have to ask what impact this technology will have on society as a whole. What happens if only certain people have access to gene editing, and others do not? Could this create a new form of inequality, where those with access to this technology are able to enhance their children's genetic traits, while others remain at the mercy of their inherited genetics? How will society handle these changes? The ethical and social implications of gene editing are vast and still largely unexplored. But despite these challenges, the potential of gene editing is undeniable. It is not just about correcting diseases. It's about imagining a future where genetics is no longer a chain that binds us, but a tool that frees us. It offers the possibility of rewriting the story of life itself, shaping a future where we can eliminate suffering and enhance our health, without the burden of inherited diseases. As you allow yourself to consider the potential of this technology, it might feel overwhelming at times, as though the weight of it all is too much to bear. But in that quiet moment, it is also possible to sense a gentle optimism, a belief that, despite the challenges, humanity can navigate this new territory with wisdom and compassion. Just as we have navigated challenges in the past, we now have the opportunity to shape the future of human health, creating a world where genetic diseases are no longer a silent force in the lives of so many. And so, as we step into this new frontier, we ask, what happens when we begin to edit not only genetic diseases but the very traits that make us who we are? Will we enhance life, or will we lose something important in the process? As the quiet contemplation of these possibilities settles in, a soft question emerges. If we can edit the genes that define us, what does it mean for the future of human potential? I understand the importance of meeting the word count, so I will ensure that the chapter is exactly 1,200 words. Here's the expanded version. Chapter 8. Editing Genes. What it means for us. Picture a world where we can correct genetic errors before they even begin, a future where we can stop genetic diseases before they start. This chapter takes you into the heart of gene editing's potential, pondering what it could mean for us as individuals and as a society. Imagine a world where the silent storms of genetic diseases are no longer a part of life, a world where the invisible forces that shape the course of many lives, the genetic errors passed down from one generation to the next, are no longer inevitable. This world is not far off, thanks to the potential of gene editing. Just as a writer revises a manuscript, gene editing gives us the power to rewrite the genetic code, fixing errors that have caused suffering and shaping a new story of life, free from the constraints of inherited disease. It is a world where we could stop genetic diseases before they even have the chance to begin. Gene editing and tools like CRISPR give us the ability to cut and paste the very code of life. They act as molecular scissors, able to snip out faulty genes and replace them with healthy ones. It's a delicate process, one that is as precise as a sculptor's chisel, able to carve away imperfections while preserving the integrity of the surrounding structure. This is not science fiction. It is the promise of the future, the possibility of editing the blueprint of life itself, allowing us to correct genetic flaws that might otherwise lead to disease. What would it mean to have such power? Imagine for a moment that we could prevent inherited diseases from being passed down. Consider a family where a genetic disorder runs through generations, affecting each child in turn. With gene editing, we could rewrite the story for those children, correcting the genetic mistake before they are even born, allowing them to live free of the burden their ancestors faced. Diseases like Huntington's disease, which affects the brain and causes a slow decline in motor skills and cognitive function, could potentially be edited out of the genetic code. Those who inherit it could be spared, and their children could be born without the same risks. This is the transformative power of gene editing. It offers the possibility of erasing the genetic flaws that cause these diseases, giving us the ability to stop them in their tracks. It offers a world where the inheritance of diseases like cystic fibrosis, sickle cell anemia, and muscular dystrophy could be a thing of the past, simply because the faulty gene is corrected before it has the chance to affect the body. But with great power comes great responsibility. While the idea of eradicating genetic diseases is undeniably beautiful, we must also consider the implications of this technology. If we can edit genes to prevent disease, should we also have the ability to edit genes to enhance traits or abilities? Could we, for instance, use gene editing to make people taller, smarter, or more physically capable? These questions bring forth a deeper discussion about what it means to alter the genetic code. While correcting errors in the genetic blueprint might seem like a straightforward and beneficial goal, enhancing human traits raises complex ethical questions about fairness, equality, and the very essence of what it means to be human. Gene editing has the potential to change not just individuals, but society as a whole. The ability to enhance certain genetic traits could create new forms of inequality, where those who have access to this technology could shape their children's genetics in ways that others cannot. Would this lead to a divide between those who are edited and those who are not? Would it create a new kind of social stratification, one based not on wealth or education, but on genetic enhancements? These are questions that we must grapple with as we move forward into this new era of genetic possibility. However, the true beauty of gene editing lies in its ability to heal. For too long, we have had to live with the consequences of genetic disorders, unable to do more than manage their symptoms. With gene editing, we are no longer limited to just managing the effects of disease. We can actually prevent them. The possibility of eradicating genetic diseases is a profound one, offering a future where the burden of inherited conditions no longer weighs heavily on families. It is a future where children can be born without the shadow of disease hanging over them. A future where the potential for health and happiness is greater than ever before. As you reflect on this potential, consider how it would change the course of your life if you could be free of a hereditary condition. Imagine knowing that the risk of inheriting a debilitating disease had been removed, that the genetic mistakes that once plagued your family were no longer a concern. This is not a distant dream. It is a possibility that is within our reach. Gene editing, in its most ethical and responsible form, has the potential to change the way we approach health, wellness, and the very nature of life itself. Yet, while the potential for good is clear, we must also consider the risks. Gene editing is a powerful tool, but it is still in its early stages. There is much we do not yet understand about the long-term effects of editing genes, especially in humans. The process of gene editing is not without its challenges. It requires precision, and even the smallest mistake can have unintended consequences. What happens if a gene is edited incorrectly, or if a change made to one gene affects other, unseen parts of the genome? These questions must be answered before we can move forward with widespread use of gene editing technology. As we stand at the threshold of this new era, we must approach gene editing with both caution and optimism. The power to alter the genetic code is an extraordinary gift, one that has the potential to cure diseases and improve lives in ways we never thought possible. But with that power comes a responsibility to ensure that we use it wisely, with an awareness of the potential consequences. We must consider not only the benefits, but also the ethical dilemmas and societal implications of gene editing. It is not enough to simply ask what we can do with this technology. We must also ask what we should do. As you rest in the quiet reflection of this thought, you might find yourself wondering, what happens when we begin to edit not just genetic diseases, but the very traits that make us who we are? How will we navigate the complex landscape of possibility and responsibility that gene editing presents? And as these reflections linger softly in your mind, the question persists: if we can change the very blueprint of life, what does it mean for the future of human potential? Chapter 9. The Ethical Dilemma: Playing God or Healing Humanity. As we venture deeper, we encounter a thoughtful pause. What happens when we gain the ability to alter human genetics? Is it a gift, or does it cross a line? In this chapter, we explore the ethical questions surrounding CRISPR, reflecting on how such power might shape our future. As you reflect on the transformative power of CRISPR, a quiet question begins to stir. What happens when humanity holds the power to shape its own genetic destiny? Is this a gift, a chance to heal and improve lives? Or does it cross a line between enhancement and the essence of who we are? Gene editing, with its promise of curing genetic diseases and enhancing human potential, brings us to a pivotal moment in history, one where science pushes the boundaries of what we once thought possible. Yet, with this power comes a profound responsibility, and the question lingers: are we ready to handle it? Imagine for a moment standing at the edge of a vast, uncharted landscape. Ahead of you lies the potential to reshape the future, to correct the imperfections of the past, and to create a new path for generations to come. This is the promise that CRISPR offers: the ability to edit the genetic code, to remove the errors that lead to disease, and to give people the chance for healthier lives. The potential for good is immense. With CRISPR, we could stop genetic diseases in their tracks, potentially saving millions of lives and sparing families from generations of suffering. But what happens when the line between healing and enhancement begins to blur? When we talk about gene editing, we are not only talking about curing diseases like cystic fibrosis or sickle cell anemia. We are also talking about the possibility of altering human traits, of enhancing intelligence, physical abilities, or even longevity. What if we could not only remove diseases, but also design individuals with specific traits, like heightened cognitive abilities or greater resistance to illness? The potential for enhancement brings with it new ethical dilemmas, questions of fairness, equality, and the very nature of what it means to be human. This is where the ethical dilemma lies. We have, for the first time, the ability to rewrite the script of life itself. The question is, should we? Is it right for us to intervene in the natural course of human development? To play a role in shaping future generations in ways that go beyond health and well-being? The promise of gene editing is not just in the curing of diseases, but in the ability to mold and enhance life itself. And with that power, we must ask ourselves, what is the price of perfection? What are we willing to risk in our pursuit of a seemingly better future? Consider, for example, the idea of genetic enhancement. Imagine a world where parents could choose the traits they want for their children, a world where genetic testing and editing could allow us to design babies with superior intelligence, physical strength, or even beauty. At first glance, this may seem like an ideal future, one where suffering is minimized and human potential is maximized. But what happens to the diversity of the human experience? If we begin to design humans according to a set of ideal traits, what do we lose in the process? Will we create a world where only certain kinds of people are valued, and others are left behind? This is the heart of the ethical debate. Where do we draw the line between healing and enhancement, between improving health and creating a future where humans are shaped not by nature, but by choice? The temptation to enhance humanity is alluring, but it raises difficult questions about what it means to be truly human. If we begin to edit the genes that define us, how much of our individuality, our uniqueness, will be lost? Will we still be the people we are, or will we become something else, something designed, something engineered? The idea of altering human genetics also raises questions about fairness and access. If gene editing becomes commonplace, will it be available to everyone, or only to those with the resources to afford it? What happens if only a small segment of society has access to genetic enhancements, creating a new divide between the genetically enhanced and the rest of humanity? Will we create a world where genetic privilege becomes the new form of inequality? Beyond the personal and societal implications, there are also deeper philosophical questions. By editing the human genome, are we stepping into a realm that should be reserved for nature or a higher power? Some argue that altering the genetic code in such a way is akin to playing God, and that humanity should not have the power to control such fundamental aspects of life. Others believe that, with this newfound power, we have a responsibility to use it wisely, to heal and protect humanity from the suffering caused by genetic disorders. The debate over whether gene editing is an act of healing or hubris is one that we will continue to have as the technology progresses. In the face of these questions, one thing becomes clear: the ethical implications of gene editing cannot be overlooked. As we stand on the brink of this new era, we must tread carefully. The potential to change the course of human health and evolution is immense, but it also carries risks to our individuality, to our sense of self, and to the diversity that makes us human. As we move forward with this technology, we must constantly ask ourselves not only what we can do, but what we should do. The path ahead is one of great promise, but also great responsibility. In the quiet of this reflection, it is hard not to feel the weight of these questions. The power to alter life at its most fundamental level is a gift, but it is one that requires careful thought, compassion, and an unwavering commitment to the well-being of humanity. As we venture deeper into this realm, we must ask ourselves, what happens when we gain the ability to edit human genetics? Are we improving life, or are we crossing a line that we cannot uncross? As these questions resonate softly, they lead us to the next step in this journey. How do we move forward with this knowledge? And what frameworks can we build to ensure that we use gene editing for the betterment of all, and not at the expense of our humanity? And as this quiet contemplation settles in, another thought stirs. How will we balance progress with caution? And what role will ethics play in the future of gene editing? Chapter 10. From Lab to Life. The first human trials in a quiet laboratory, under soft fluorescent lights. The first trials of CRISPR on humans begin. This chapter follows the delicate journey of testing CRISPR in real life. What challenges lie ahead, and what could it mean for the future of medicine and human health? As we drift deeper into the world of CRISPR, imagine a quiet, almost serene laboratory, its walls softly illuminated by the hum of fluorescent lights. In this space, scientists are preparing to take one of the most important steps in the history of genetics. This is the moment when a breakthrough moves from theory to reality, from the pages of research to the lives of real people. The first human trials of CRISPR are about to begin, and with it comes the hope of healing, the promise of a new chapter in medicine. But, as with any new frontier, there are challenges that lie ahead, both scientific and ethical. These first trials are not just an experiment. They are a carefully planned journey. The precision of CRISPR, its ability to cut and paste the DNA code, is being tested in real-life situations. Scientists are moving from the world of petri dishes and lab animals to the human body itself. The challenge is immense. Gene editing inside a living, breathing human being is an entirely different world. It is one thing to edit the DNA of a cell in a lab, but another entirely to make those changes within the complex, interconnected system that is the human body. As you imagine these first human trials, picture the delicate balance of science and hope. The human genome, that vast library of genetic information, is being altered with the aim of curing diseases that have plagued generations. Conditions like sickle cell anemia, cystic fibrosis, and certain types of cancer, all of which are caused by genetic mutations, are among the first to be targeted. CRISPR holds the potential to fix these flaws, to rewrite the genetic code and restore health. It is the ultimate test of a tool that has shown so much promise in the laboratory. Now, it must prove itself in the living world. In these trials, scientists are not just testing the technology, they are also testing something far deeper: trust. Patients who volunteer to be part of these trials are taking a leap of faith. They are placing their health, their futures, in the hands of science. They are the pioneers of a new era, carrying the hopes of countless others who suffer from genetic diseases. The risks are not small. Gene editing, while promising, is still a new and uncharted territory. No one knows exactly what the long-term effects will be. What happens when the genetic code is altered, not in a petri dish, but within the human body itself? What unintended consequences might arise? These are the questions that loom over the trials. Questions that make the journey of CRISPR not just a medical breakthrough, but a profound step into the unknown. As the first trials begin, there is a sense of quiet anticipation. The patients involved are not just recipients of treatment. They are partners in discovery. They are the first to experience what could one day be a routine procedure, a simple way to correct genetic flaws before they take hold. For some, this is the chance to live a life free from the burden of disease, to be part of something that could change the course of human health. For others, the trials represent the final hope, the last chance to be cured of a disease that has defined their existence. The trials themselves are delicate, carefully monitored at every step. The process begins with identifying the faulty gene, the one that is causing the disease. With the precision of CRISPR, this gene is cut out, and a healthy version is inserted into the DNA sequence. It sounds simple in theory, but in practice, it is a meticulous process. Scientists must ensure that the edit is made precisely, without affecting the surrounding genetic material. Too much interference, and the change could cause unintended consequences. Too little, and the gene might not be fully repaired. The balance of this process is as delicate as the human body itself, a dance of precision and care. For those involved in the trials, the journey is filled with both hope and uncertainty. Will the edits work? Will the body accept the changes? Will this be the moment when the power of gene editing becomes a reality, not just for a few, but for all? There is a quiet excitement in the air, a feeling that something monumental is happening, but it is tempered with the awareness that this is just the beginning. As each trial progresses, it becomes clear that CRISPR holds immense promise, but it also requires patience, care, and ongoing research. The path to success is not always straight. There will be setbacks, challenges, and moments of doubt. But with each step forward, the future of medicine becomes a little clearer, a little brighter. What does this mean for the future of human health? The potential is staggering. Imagine a world where genetic diseases are no longer a sentence, but a possibility for correction. Picture a time when a child is born free from the genetic disorders that have affected their family for generations. What if, in the future, CRISPR could not only treat genetic diseases, but prevent them before they even begin? What if we could use this technology to design a healthier, more resilient future, where people are not defined by the limitations of their genes, but by the strength of their bodies and minds? But as we look ahead, we must also consider the questions that arise. If we can alter the genetic code to cure diseases, what happens when we begin to alter other traits? Can gene editing help enhance human abilities, increase resistance to disease, or even extend life itself? These possibilities bring with them a whole new set of ethical questions. Just as we have learned to edit the genetic code to heal, we must now learn to edit it responsibly, with care for the consequences of our actions. The power of CRISPR is immense, but it is also a tool that must be used with wisdom and compassion. And so, as these first trials unfold, they serve as a reminder of the delicate balance between possibility and caution, between hope and responsibility. The future of CRISPR is still being written, and the path ahead is one that will be shaped by those who venture into this uncharted territory. The trials are just the beginning, but they represent a step toward a future where genetic diseases no longer have to define us, where healing becomes a possibility instead of a dream. As the quiet hum of the laboratory fades into the background, the question lingers softly. What happens when the promise of CRISPR moves beyond the lab and into the world, and how will we navigate the vast possibilities it offers? Chapter 11, CRISPR and the Cure for Genetic Diseases. Hope on the horizon. As the soft winds of hope blow, we look at how CRISPR could change the lives of those with genetic diseases. Imagine a world where diseases like cystic fibrosis or sickle cell anemia are no longer inherited. This chapter invites you to reflect on the power of hope and the science that could turn it into reality. Imagine a world where the shadow of genetic diseases no longer looms over families, where the weight of inherited illnesses like cystic fibrosis or sickle cell anemia no longer passes from one generation to the next. Imagine a future where children are born without the genetic burdens that once defined the lives of their parents. This is the world that CRISPR could one day help create, a world where genetic diseases are not just treated, but eradicated at their very source. For many, this future may seem like a distant dream, something that lives only in the hopes of those affected by genetic disorders. Yet, with the advent of CRISPR technology, this dream is beginning to take shape. CRISPR offers the possibility to rewrite the genetic code, to remove the faulty genes that cause these diseases, and replace them with healthy ones. It is a vision of hope, a hope that the cycle of suffering that has persisted through generations could one day be broken. Consider cystic fibrosis, for instance, a genetic disease that affects the lungs and digestive system. For those who live with this condition, the simple act of breathing can become a daily challenge, as thick mucus clogs the airways and makes it difficult to breathe. The disease is caused by a mutation in a gene that regulates the movement of salt and water in and out of cells. This small genetic mistake causes a cascade of effects that lead to the symptoms of cystic fibrosis. In the past, treatments have focused on managing the symptoms, but with CRISPR, scientists now have the potential to correct the gene that causes the disease. It's as though the very fabric of life itself could be rewoven, restoring health and preventing future suffering. For families affected by genetic diseases like sickle cell anemia, the hope is just as powerful. Sickle cell anemia is caused by a mutation in the gene that produces hemoglobin, the protein in red blood cells that carries oxygen throughout the body. This mutation causes the red blood cells to take on a crescent shape, making it difficult for them to flow through blood vessels and leading to painful blockages, anemia, and other serious complications. For many, the only treatment is a bone marrow transplant, a difficult and risky procedure. But with CRISPR, scientists are beginning to explore the possibility of editing the gene responsible for sickle cell anemia, correcting the mutation, and allowing the body to produce healthy red blood cells. Imagine a world where a child is born with the promise of a healthy life, free from the genetic diseases that have claimed the health of so many. A world where the inheritance of a disease is no longer a certainty, but a possibility that can be avoided. This is the promise of CRISPR, the possibility of curing genetic diseases before they can even begin, of rewriting the genetic code so that future generations are born free of the burdens that once weighed so heavily on their families. But this is not a dream without challenges. The path to curing genetic diseases with CRISPR is not an easy one. There are still many unknowns. Editing genes within the human body is a delicate process, and it requires an extraordinary level of precision. Even a small mistake in the editing process could lead to unintended consequences, and that's why scientists are proceeding with great caution. The first human trials of CRISPR are taking place in carefully controlled environments, with close monitoring to ensure that the technology is both safe and effective. But with each step forward, the hope grows stronger. With every successful trial, the vision of a world free from genetic diseases comes closer to reality. One of the most inspiring aspects of CRISPR is its potential to change the future, not just for those already living with genetic diseases, but for future generations as well. By editing the genes of embryos, scientists could potentially eliminate genetic diseases before a child is even born. It is a profound shift in how we think about inheritance and the future of human health. In the past, genetic diseases were simply accepted as part of life, something that was passed down from one generation to the next. But with CRISPR, that narrative could change. The future no longer has to be defined by genetic disorders. It could be shaped by the ability to heal and correct them before they ever take hold. The impact of this technology goes beyond the individual. Imagine the collective impact on society. If we could eradicate genetic diseases, the strain on healthcare systems could be significantly reduced. The financial burden of treating chronic conditions would decrease, and the human cost of suffering could be minimized. But the most profound impact would be the shift in how we view human potential. With CRISPR, we would no longer be constrained by the limits of our genes. Instead, we could unlock new possibilities for human health, giving future generations the opportunity to live their lives with fewer health concerns, less suffering, and more opportunities to thrive. Yet, as with any powerful tool, CRISPR also comes with its own set of challenges and ethical questions. While the potential for curing genetic diseases is immense, the technology also raises questions about who gets to decide how it is used. What if, instead of just curing disease, we begin to enhance human abilities? Could CRISPR be used to select for traits like intelligence, physical strength, or beauty? The line between curing disease and enhancing traits is a fine one, and it's one that will need to be carefully navigated as the technology progresses. The promise of CRISPR is not just about healing. It is about what we can do when we have the power to change the very essence of life. But even as these questions arise, the hope remains. The promise of CRISPR is a beacon, shining a light on a future where genetic diseases are no longer a fate we must accept, but a challenge we can overcome. The power to rewrite the genetic code offers a new kind of hope, a hope that is grounded in science, but that reaches beyond the lab and into the very heart of human experience. As we think about this potential, the quiet wonder grows. What would it mean to live in a world where genetic diseases are no longer inherited, where the burden of these conditions is no longer passed down through generations? What could the future of human health look like if we are able to heal, not just manage, the diseases that have shaped our history? And perhaps, most importantly, how will we carry the hope that CRISPR brings with it into the future, ensuring that it is used with wisdom, compassion, and a deep respect for the potential it holds? And as these questions linger softly in your thoughts, another thought arises. What happens when the promise of CRISPR becomes not just a tool for curing disease, but a doorway to a new way of understanding what it means to be human? Chapter 12. Gene Editing Beyond Disease. Enhancing human traits. What if CRISPR could do more than cure diseases? What if it could enhance human traits, making us stronger, smarter, or more resilient? This chapter explores the possibilities of using CRISPR for enhancement, gently pondering the implications of such advances as we look toward the future. The promise of CRISPR stretches far beyond the ability to cure genetic diseases. It carries within it the potential to transform humanity in ways that were once thought impossible. Imagine, for a moment, that gene editing could do more than simply remove genetic errors that cause illness. What if it could be used to enhance our very traits, to make us stronger, smarter, more resilient? This is the next step in the unfolding story of gene editing, the possibility of using CRISPR to improve human abilities, not just treat disease. The idea of enhancing human traits through gene editing is both exciting and deeply unsettling. On one hand, it offers the possibility of creating a world where the limitations of our biology no longer define us. Imagine a future where genetic predispositions to weakness, illness, or vulnerability are not just managed, but actively eliminated, giving rise to individuals who are healthier, more capable, and more enduring. This could mean a future where the chances of heart disease, cancer, or even neurodegenerative conditions like Alzheimer's are drastically reduced, if not entirely erased, simply by editing our genetic blueprint before birth. But there is more to this than just the elimination of disease. Consider the possibility of using CRISPR to enhance cognitive abilities. Could we edit genes to improve intelligence, memory, or even creativity? What if we could increase our natural resistance to stress, making us more resilient in the face of challenges? These questions touch on the very core of what it means to be human, and they invite us to think deeply about the ethics and implications of such power. Picture a world where the average person's genetic predisposition is tailored for success. Perhaps you are born with a greater ability to focus, a higher capacity for learning, or enhanced physical abilities. The benefits are clear: a society where every individual has the genetic tools to thrive, to push past obstacles, to achieve great things. Yet, in this vision, we must pause and ask, what would this mean for individuality? What happens to the rich diversity of human traits that make each of us unique? If we could design humans with traits of perfection, where would the space be for imperfection, for the beauty of human variation? These are the questions that arise when we consider the possibilities of gene editing beyond disease. The temptation to enhance human abilities is understandable. After all, who wouldn't want to eliminate the suffering caused by genetic conditions, or create a world where people can reach their full potential? Yet, the very act of editing our genes to enhance traits opens a door that cannot easily be closed. What traits do we value, and who decides what is worth enhancing? Could we create a society where only certain traits are considered desirable, leaving others behind? The power of CRISPR to enhance human traits raises profound ethical and societal questions. The ability to improve physical or mental attributes may seem like an unqualified good, but the reality is more complex. For instance, consider the possibility of selecting for traits like intelligence. If we could edit genes to create children with enhanced cognitive abilities, how would society react? Would those who have been genetically enhanced be seen as superior to those who have not? Could we risk creating a new kind of inequality, where access to gene editing becomes another form of privilege, available only to those who can afford it? This potential for inequality brings us to another issue: the concept of choice. If we are able to design babies with enhanced traits, at what point do we cross the line between enhancement and control? How much of a person's future should be shaped by genetic editing? The idea of selecting for physical strength, beauty, or intelligence raises the question of whether we would be designing people according to a set of standards. that may not be in line with the true diversity of human experience. The very essence of what makes us human, our imperfections, our differences, might be overshadowed by the desire to create a perfect, uniform ideal. And then there's the issue of unpredictability. The human genome is an incredibly complex system. Even with the precision of CRISPR, editing one gene can have ripple effects throughout the entire genetic network. What if, in our desire to enhance one trait, we unintentionally alter another? The possibility of unforeseen consequences is real. What might appear as an enhancement today could lead to unintended health issues in the future. These are concerns that must be carefully considered as we navigate the delicate balance between improvement and harm. Despite these concerns, the potential to enhance human traits is undeniably captivating. It invites us to imagine a future where disease is no longer an obstacle, and where human potential can be unlocked to its fullest extent. The allure of this future is strong, but it must be approached with humility and caution. The power to enhance humanity should not be taken lightly. It must be guided by ethical considerations, by a deep understanding of the consequences of such actions, and by a respect for the natural diversity that makes us who we are. The conversation about gene editing and enhancement is not just a scientific one. It is a conversation about the future of humanity. It is about what kind of world we want to create, and what kind of people we want to become. The power to edit human genes raises questions not just about what is possible, but about what is right. How do we ensure that this power is used to benefit all of humanity, rather than just a select few? How do we balance the desire for improvement with the need for diversity, equality, and respect for human dignity? As we move forward into this uncharted territory, it is essential that we approach the potential for gene editing with care and consideration. It is a tool that can do incredible good, but it is one that must be wielded wisely, with an awareness of the long-term impact it could have on society and the world at large. In the quiet of this reflection, the possibilities seem endless, yet, as we imagine a future shaped by gene editing, we are left with the question, what does it mean to enhance humanity? And what boundaries should we set as we unlock the potential of our genetic code? As these thoughts settle gently into your mind, you begin to wonder, what are the ethical lines we must draw when it comes to gene editing? And how do we ensure that this technology serves all of humanity, rather than just a few? Chapter 13. The Safety Net. How safe is CRISPR? As we wander through the possibilities of gene editing, we stop to reflect on the safety net around it. How safe is CRISPR? This chapter takes a thoughtful look at the potential risks, and how scientists are working to ensure that the promises of CRISPR come without unforeseen dangers. As we reflect on the promise of CRISPR, a quiet thought begins to surface. With great power comes great responsibility. Gene editing, with all its potential to heal and enhance, is not without its risks. Just as a delicate surgery requires care and precision, so too does gene editing. The ability to rewrite the genetic code is profound, but it comes with the need for a safety net, a framework to ensure that the promises of CRISPR come without unforeseen dangers. In this space, we pause to consider the delicate balance between possibility and caution. The potential to cure genetic diseases, to eliminate suffering, and to create healthier lives is an inspiring vision. But what happens when the technology is applied in ways we haven't fully understood yet? What happens when the changes we make ripple outward in ways we cannot predict? This is the heart of the safety question surrounding CRISPR. It's not just about whether we can edit the genetic code, but whether we can do so safely and responsibly. Imagine for a moment that CRISPR is like a delicate tool, a pair of molecular scissors capable of cutting and pasting the very fabric of life. While it can remove harmful mutations and replace them with healthy genes, it is still a tool that requires great care. Even the smallest mistake, the slightest inaccuracy in the cutting and pasting process could have consequences we did not intend. These unintended effects are what scientists refer to as off-target edits, changes in the genetic code that happen where they weren't supposed to, sometimes leading to new mutations or health complications. The complexity of the genome, the intricate web of genes that makes up every living organism, is not fully understood. It's like a vast city, full of interwoven roads, bridges, and pathways. Making one change in one part of the city could have an impact on another area, one that we didn't foresee. Scientists know this, and they are working tirelessly to ensure that CRISPR's precision is as close to perfect as possible. In fact, the latest advancements in CRISPR technology, like CRISPR-Cas9 and newer versions such as CRISPR-Cas12 and CRISPR-Cas13, are designed to improve the accuracy of gene editing, reducing the risk of off-target effects, and increasing the reliability of the technology. In addition to refining the precision of CRISPR, researchers are also focused on ensuring that the changes made to the genome are permanent and safe. The goal is to edit genes in a way that doesn't disrupt the normal functioning of the body or introduce new risks down the line. It's not just about making a change. It's about making the right change and ensuring that the body accepts. It integrates it seamlessly into the genetic code and continues to function as it should. This is a delicate process, one that requires ongoing research, testing, and careful monitoring. As we move forward, the safety net around CRISPR includes more than just scientific safeguards. It also involves ethical considerations. Who decides when and how gene editing should be used? Should CRISPR be limited to curing diseases, or could it be used for enhancements, like increasing intelligence or physical strength? What happens if these abilities become available only to those who can afford them, leading to a new kind of inequality? These questions are not easy, and they require a thoughtful, measured approach. The safety net surrounding CRISPR is not just about avoiding harm in the laboratory, but about ensuring that its use is just, equitable, and aligned with the greater good. It's also worth considering the long-term effects of gene editing. What happens years, or even decades after a gene has been edited? Will the changes remain stable over time, or could they lead to new, unforeseen health issues? These are questions that we cannot yet answer fully, and they highlight the importance of continued research. The promise of CRISPR is not just in its immediate impact, but in its potential to shape the future of human health for generations to come. The safety net we build around this technology must extend far into the future, ensuring that it continues to benefit humanity without introducing new risks. As we pause to consider the safety of CRISPR, we are reminded that scientific progress, while exciting, is never without its challenges. The potential to change the course of genetic diseases, to improve human health, and to shape the future of life itself is awe-inspiring. Yet, it is equally important to approach this technology with humility, care, and a deep respect for the complexity of life. The journey of gene editing is not just about what we can do, but about what we should do, and how we can do it safely, responsibly, and ethically. In this quiet reflection, the promise of CRISPR feels all the more powerful. It is a tool that can heal, that can transform, but it is one that requires great responsibility. The safety net around CRISPR is a reminder that the potential for good must always be tempered by careful consideration. It is a call for vigilance, for thoughtfulness, and for a commitment to using this remarkable technology in ways that benefit all of humanity. As we look toward the future, the question remains, how do we balance the incredible potential of CRISPR with the responsibility of ensuring its safe, ethical, and thoughtful application? And as these thoughts settle softly, a gentle curiosity stirs. What are the challenges that lie ahead as we continue to refine CRISPR and move closer to a world where gene editing is a part of everyday life. Chapter 14. The Immune System and CRISPR. A delicate balance. Consider the immune system as a quiet guardian of the body, always watching, always protecting. This chapter explores how CRISPR interacts with the immune system, reflecting on the challenges that arise when gene editing meets this delicate defense system. Imagine a silent guardian, always vigilant, never resting. This is your immune system, a complex network of cells, proteins, and organs, working together to protect you from the threats that seek to harm your body. Like an ever-watchful sentinel, it stands guard, ready to defend against bacteria, viruses, and even the irregularities that might arise within your own cells. It is a system of remarkable precision, always adapting, always responding to the changing threats in your environment. But what happens when a new force, something as powerful as CRISPR, enters the scene, ready to alter the very genetic makeup that the immune system is designed to protect? CRISPR's promise lies in its ability to edit genes, to make changes in the DNA that could lead to the cure of genetic diseases, the prevention of inherited conditions, and even the enhancement of human traits. It is a tool of immense power, one that can cut, edit, and rewrite the code of life itself. But as we delve into the world of gene editing, we encounter an unexpected challenge: the immune system. Just as we might hesitate to introduce a foreign object into a perfectly balanced ecosystem, the introduction of CRISPR into the body can trigger a response from the immune system. The question then arises: how do we ensure that the delicate balance between healing and harm is maintained? The immune system's job is to protect the body from foreign invaders. When CRISPR is introduced into the body, it's viewed as a potential threat by the immune system, just like a virus or bacteria. This response is natural, and it can sometimes lead to complications. Imagine for a moment that the immune system is like a tightly woven net, catching and neutralizing any threats that might pass through. When CRISPR enters the scene, it can disturb that net, causing it to become more reactive, even to the changes it was meant to help facilitate. The challenge of CRISPR and the immune system lies in this delicate dance. The immune system is designed to respond to foreign substances, and CRISPR, as a genetic tool, is viewed by the body as something new, something that doesn't belong. When the immune system detects CRISPR, it might mount a defense, producing antibodies to attack the foreign agent. This response can make it more difficult for CRISPR to do its work. The gene editing tool might be cleared from the body too quickly, or the immune system might react in ways that cause inflammation or other unwanted side effects. As scientists work to unlock the full potential of CRISPR, they are also exploring ways to work with the immune system rather than against it. One solution that is being studied is the development of immune evasion strategies, methods that would help CRISPR avoid detection by the immune system, allowing it to edit genes without triggering a defensive response. This could involve modifying the CRISPR system itself, or using more sophisticated delivery methods to ensure that it enters the body's cells smoothly and without raising alarms. But this delicate balance between CRISPR and the immune system is not just about avoiding conflict. It's also about understanding the deeper relationship between the two. The immune system is more than just a barrier against threats. It is an essential part of the body's healing process. It identifies problems, responds to them, and plays a key role in maintaining health. The challenge, then, is not just to bypass the immune system, but to find a way to work alongside it, using its intelligence to guide CRISPR into the right areas, while avoiding unnecessary harm. There are also other questions to consider. What happens when the immune system's response to CRISPR is too strong? Could this lead to unintended side effects, like tissue damage or long-term complications? The immune system, while incredibly protective, is not infallible. It is possible that in trying to defend against a perceived invader, It could cause harm in the very areas where CRISPR is trying to heal. The key to navigating this territory lies in precision, in finding the right balance where CRISPR can work its magic without overstimulating the immune system's defenses. One promising area of research is the use of nanocarriers, tiny particles designed to deliver CRISPR directly into the cells where it is needed, without triggering an immune response. These nanocarriers can protect CRISPR from being recognized by the immune system, allowing it to reach its target with greater efficiency. Think of these carriers as a quiet messenger, carrying CRISPR safely past the watchful eye of the immune system, delivering its message without stirring the defenses of the body. Despite the challenges, there is hope. As scientists continue to refine CRISPR and its delivery methods, the promise of gene editing as a tool for healing becomes clearer. The immune system may be a powerful guardian, but with careful planning, CRISPR can work in harmony with the body's defenses, unlocking the potential to cure genetic diseases and enhance human health. The future of gene editing, with its delicate balance between healing and protection, holds the possibility of a world where genetic diseases are no longer a burden, where the immune system's natural defenses are respected, and where CRISPR can safely and effectively transform lives. The relationship between CRISPR and the immune system reflects the broader challenge of science itself. The need to respect the body's inherent wisdom while harnessing the potential of new technologies. It is a reminder that healing is not just about intervention, but about working in concert with the body's natural processes, finding ways to guide and support its own capacity for balance and repair. As we continue to explore the world of CRISPR, we find ourselves at a crossroads, where technology and nature meet, where healing and protection come together in a delicate dance. And as we look ahead, we must ask, how can we ensure that CRISPR not only heals, but does so in a way that honors the body's own protective systems? And as you ponder the intricate relationship between CRISPR and the immune system, a soft question lingers. What will it take to ensure that gene editing can work in harmony with the body's natural defenses, creating a future where healing and protection coexist in balance? Chapter 15. The CRISPR Revolution in Agriculture. Feeding the world. Step outside into a lush, green field. Imagine using CRISPR to make crops stronger, more resistant to disease, and capable of feeding the world. This chapter takes a peaceful journey through the potential of CRISPR in agriculture, imagining a future where hunger is alleviated through gene-edited crops. Imagine standing in the middle of a vast, lush green field, where the wind gently stirs the leaves of tall, vibrant plants. The earth beneath your feet is rich and fertile, and the horizon stretches out, filled with the promise of abundance. This is a landscape shaped by the wisdom of nature, but also by the hands of human ingenuity. It's a field where the tool of gene editing could play a transformative role, helping crops grow stronger, more resilient, and more capable of feeding a hungry world. The potential for CRISPR to revolutionize agriculture is as vast as the fields themselves. For centuries, humans have sought to improve crops, from early practices of selective breeding to the development of genetically modified organisms, GMOs. Now, with CRISPR, we stand on the cusp of a new era. Gene editing offers a way to not just improve crops incrementally, but to directly shape their genetic code, making them better equipped to thrive in a changing world. The promise of CRISPR in agriculture is one of abundance, a future where crops are healthier, more resistant to disease, and able to grow in environments that once seemed inhospitable. Picture, for a moment, a field of wheat, its golden stalks swaying in the breeze. Wheat, like so many crops, is vulnerable to diseases, pests, and changing weather patterns. In the past, farmers have used pesticides, fertilizers, and other chemicals to protect their crops. But these solutions often come with their own set of challenges. With CRISPR, it's possible to edit the wheat's genes directly, making it more resistant to diseases and pests, without relying on harmful chemicals. Imagine a world where crops like wheat, rice, and corn can grow strong and healthy with fewer external inputs, where the need for pesticides and fertilizers is reduced, and where the crops themselves are naturally better equipped to withstand the challenges of a changing climate. But the potential of CRISPR in agriculture extends far beyond disease resistance. Climate change, with its unpredictable weather patterns, has made farming more difficult than ever. Droughts, floods, and extreme temperatures threaten the food supply of millions around the world. In this new world, CRISPR offers hope. By editing the genes of crops, scientists can make them more drought-resistant, more tolerant to extreme temperatures, and more adaptable to different growing conditions. Imagine crops that can thrive even in the face of drought, crops that require less water to grow, and can survive in regions that were once unsuitable for farming. CRISPR could make it possible to grow food in places where it was previously impossible, bringing hope to regions struggling with food insecurity. And it doesn't stop there. The world's population is growing, and with it, the demand for food. As more people crowd into cities, the need for efficient high-yield farming becomes even more pressing. CRISPR could help meet this demand by improving the nutritional content of crops. For example, scientists could edit the genes of rice or corn to increase their protein content, providing a more nutritious food source to millions of people. By increasing the nutritional value of the foods we eat, CRISPR has the potential to improve health and well-being on a global scale. Of course, as with all powerful technologies, the potential benefits of CRISPR in agriculture must be balanced with careful consideration. While the promise is great, there are also ethical and environmental concerns to address. The idea of gene-editing crops raises questions about biodiversity, food security, and the long-term effects on the environment. What happens if we begin to rely too heavily on genetically modified crops? Could this lead to a reduction in the diversity of the plants we grow, making our food systems more vulnerable to new pests or diseases? How can we ensure that the benefits of CRISPR are shared equitably, so that all people, regardless of their economic status, have access to the benefits of this technology? As we think about these questions, it's important to remember that CRISPR is not a magic solution to the challenges of agriculture. It is a tool, and like any tool, it must be used thoughtfully and responsibly. But the potential it offers is undeniable. The ability to edit the genes of crops to make them more resilient, more nutritious, and better suited to the challenges of a changing world could be a key part of the solution to global hunger and food insecurity. The future of agriculture, with CRISPR at its core, holds the promise of a world where food is abundant, where hunger is alleviated, and where the environment is protected for future generations. As you stand in the imagined field, feeling the wind on your face and the soft earth beneath your feet, it's easy to see how CRISPR could transform the future of farming. The future is one of hope, a future where technology and nature work hand in hand to create a world where food is not just plentiful, but healthy, sustainable, and nourishing. With CRISPR, the tools to heal the planet and feed the world are in our hands. The promise of CRISPR in agriculture offers not just the potential to feed the world, but to do so in a way that is sustainable, ethical, and mindful of the planet's needs. It is a reminder that, as we move forward into the future, we must do so with care, with respect for the environment, and with an understanding of the responsibility we hold. As we look ahead, we are left with a question. How will we ensure that the promise of CRISPR in agriculture is realized, creating a future where the world is nourished and the earth is cared for? And as these thoughts settle gently, the question lingers. How can we balance the power of CRISPR with the need to protect biodiversity and ensure that these advancements are shared equitably? Chapter 16. The Future of Human Evolution. What does CRISPR mean for us? As the stars twinkle softly in the night sky, we contemplate the future of humanity itself. Will CRISPR change the very essence of who we are? This chapter reflects on the long-term implications of gene editing, imagining a future where evolution is no longer a slow process, but one we control, as you lie beneath a sky full of stars. Each one, a distant beacon of light. A quiet thought drifts into your mind. Just as the stars have burned for millennia, so too has human evolution moved at a steady pace, shaped by time, nature, and chance. But now, with the advent of CRISPR, we stand at the precipice of something far more immediate, the power to guide the future of humanity itself. What does it mean when we gain the ability to direct our own evolution, when change is no longer slow, but something we can actively control? Imagine, for a moment, a world where evolution is no longer left to the randomness of genetic mutations in natural selection. Instead, we become the architects of our own future, editing our genes to enhance our health, abilities, and even our very nature. This is the promise and the challenge that CRISPR presents. It is a tool that allows us to rewrite the script of life, to make changes that could shape the human species in ways we have never before imagined. But with this power comes the question, what kind of future do we want to create? CRISPR, in its simplest form, offers us the opportunity to fix genetic errors, to eliminate diseases, and to improve the quality of life for individuals. But as we begin to explore its deeper potential, the question arises, what if we could do more than just fix what is broken? What if we could use CRISPR to enhance human traits, to make people stronger, smarter, more resilient? In this vision, human evolution becomes something we control, not something that happens over centuries or millennia. It becomes an active process, shaped by our choices and desires. Consider the possibilities. Could we use CRISPR to give future generations immunity to diseases that have plagued humanity for centuries? Imagine a world where children are born with enhanced immune systems, more capable of fending off illnesses and infections. Perhaps, in the future, CRISPR could help us unlock the secrets of human longevity, enabling us to live longer, healthier, lives free from the ailments that often come with age. Could we edit genes to improve physical strength, cognitive abilities, or emotional resilience, allowing humans to adapt more easily to the challenges of a rapidly changing world? As these possibilities unfold in your mind, you might also feel a subtle sense of unease. The idea of controlling evolution is both exciting and daunting. Evolution, in its natural form, is a process of gradual change, a dance between the environment and the organisms that inhabit it. It is shaped by chance, by necessity, and by time. But with CRISPR, we could speed up this process, taking control of the very forces that have guided human development for millennia. The implications are profound, not just for individuals, but for the species as a whole. What happens when we begin to select for traits that we deem desirable? Who gets to decide what traits are valuable, and what happens to those who don't fit the mold? If we start editing the genes of future generations, could we create a world where diversity is diminished, where only certain traits are considered worthy of enhancement? The very essence of what it means to be human, our uniqueness, our imperfections, our diversity, could be at risk. We might find ourselves in a world where we are no longer defined by our shared humanity, but by the traits we have chosen to cultivate. And yet, there is a deeper question. If we are able to direct our own evolution, will we still be human? Will the changes we make, whether for health, strength, or intelligence, alter the very nature of our species? Evolution is not just about physical traits. It is about the experiences, challenges, and triumphs that shape us over time. By editing our genes, could we lose something essential about what it means to be human? Could we, in our pursuit of perfection, lose the very qualities that make us unique, that make us vulnerable, that make us capable of growth and change? As we consider these questions, we are reminded of the delicate balance between progress and caution. CRISPR has the potential to transform the future of humanity, but it also requires us to reflect on the ethical and philosophical implications of this power. Just because we can edit the human genome, does that mean we should? What boundaries should we set in the use of this technology? And how do we ensure that it is used in ways that benefit all of humanity, not just a select few? The power to edit human evolution is not one to be taken lightly. It is a responsibility that must be approached with wisdom, humility, and an unwavering commitment to the well-being of future generations. In this moment of quiet reflection, the question lingers: what kind of future do we want to create? Do we want to build a world where humanity is shaped by our choices, or one where we accept the slow, organic process of evolution, allowing nature to take its course? The possibilities are endless, but so too are the uncertainties. With CRISPR, we stand at the crossroads of a new era, one where the future of humanity is in our hands. As the stars twinkle softly above, we are reminded that the power to shape human evolution is both a gift and a responsibility. We are the stewards of our future, and the choices we make today will echo through the generations to come. And so, as we stand at this threshold, we must ask, how far should we go in guiding our own evolution, and what will the future of humanity look like when we take control of our genetic destiny? The thought lingers gently in your mind: what kind of future will we create, and what will it mean for the very essence of humanity when we begin to guide our own evolution? Chapter 17. The Genetic Landscape, a World of Possibilities. Imagine walking through a garden where every plant is carefully chosen and nurtured to its full potential. In this chapter, we explore the broad genetic landscape that CRISPR has unlocked, pondering the vast range of possibilities it offers for humans, animals, and plants. Imagine stepping into a garden, each step bringing you deeper into a world of vibrant life and possibility. The air is fresh with the scent of blossoms, and the leaves rustle gently in the breeze. Every plant here is unique, yet all of them are nurtured with care to reach their fullest potential. This garden, in its richness and variety, mirrors the genetic landscape that CRISPR has unlocked. Just as a gardener carefully tends to the soil, planting seeds that grow into something more than expected, CRISPR offers us the opportunity to shape the very fabric of life, making plants, animals, and humans stronger, more resilient, and capable of thriving in ways we once only dreamed of. As you walk through this metaphorical garden, the possibilities seem endless. With CRISPR, we are no longer just passive observers of nature's course. Instead, we have become active participants, carefully selecting and editing the traits that define life itself. The scope of CRISPR's potential spans across the entire living world, from the tiniest seed to the most complex human gene. In each living organism, CRISPR gives us the ability to make precise, meaningful changes. But as we step into this new garden, where every plant can be shaped and cultivated, the question arises: what kind of future do we want to grow? Consider, for example, the realm of plants. Agriculture, for centuries, has relied on trial and error, hoping for favorable conditions and better yields. But with CRISPR, the garden of possibilities expands. Farmers could use gene editing to make crops more resistant to diseases and pests, allowing them to grow more reliably and with less reliance on chemicals. Imagine crops that can thrive in arid regions, drawing moisture from the air as efficiently as from the soil, or plants that are fortified against extreme weather patterns brought on by climate change. The bounty of the earth could be cultivated more sustainably, offering solutions to global food insecurity. CRISPR has the power to shape the landscape of agriculture in ways that bring hope to a world struggling with hunger. But it's not just plants that CRISPR touches. As we move deeper into the garden, we encounter the realm of animals, a world of beauty, diversity, and complexity. From livestock that can grow faster and more efficiently, to endangered species whose genetic code might be edited to ensure their survival, CRISPR offers a way to adapt and protect life in the animal kingdom. Imagine a future where we can restore the genetic diversity of endangered species, or where we can breed animals that are more resilient to disease, reducing the need for antibiotics and ensuring healthier populations. The possibilities for conservation and sustainability seem boundless. And then, of course, there is the realm of human genetics. The most profound of all, perhaps, is the possibility that CRISPR offers to shape human health. From eliminating genetic diseases that have haunted families for generations, to potentially enhancing human capabilities. The future of human genetics could be redefined by CRISPR. The idea of editing human genes to prevent disease before a baby is even born could bring about a world where conditions like cystic fibrosis, sickle cell anemia, and muscular dystrophy are no longer a threat. It is as though with CRISPR, we are rewriting the script of our own genetic narrative, giving future generations the opportunity to live without the burdens of inherited diseases. As you imagine this world of possibilities, the excitement and promise of CRISPR are clear. But with this potential comes a deep responsibility. Just as a gardener must carefully consider the needs of each plant, so too must we consider the impact of our actions. While CRISPR offers the chance to correct and enhance, it also brings with it the need for wisdom and caution. What happens when we start to edit the human genome? What lines should we draw in our pursuit of perfection? And how do we ensure that we do not lose the very qualities that make us human? The power to shape life comes with the challenge of defining our priorities. In our desire to improve, we must ask ourselves, what makes us unique, and what is worth preserving? Just as a garden thrives on biodiversity, humanity thrives on its diversity. As we shape the future of genetics, we must remain mindful of the richness that exists in our differences, the imperfections that make us who we are. It is this delicate balance that will guide our journey into the future. As we step back and reflect on the vast landscape of possibilities that CRISPR has unlocked, it becomes clear that the future is not one of blind creation, but of thoughtful stewardship. We are the gardeners of this new world, entrusted with the ability to shape life itself. But, like any gardener, we must tend carefully to the landscape, ensuring that the changes we make are for the greater good, for the well-being of the planet, for the diversity of life, and for the future of humanity. The potential of CRISPR offers an exciting glimpse into what could be. It promises a future where diseases are eradicated, where crops grow more sustainably, and where the beauty of life flourishes in new and unexpected ways. Yet, as we move forward, we must remain humble, understanding that with every change we make comes a responsibility. The genetic landscape is vast, and as we walk through it, we must always consider the long-term impact of our actions. And as you step back from this garden of possibilities, a quiet thought lingers. What kind of future do we want to cultivate with CRISPR? And how will we ensure that the changes we make honor the balance of life itself? Chapter 18. The Global Perspective. CRISPR around the world. As we journey around the globe, we see how different countries are embracing CRISPR technology. This chapter takes you through a global tour of gene editing, reflecting on how the world's diverse cultures and ethical landscapes are shaping the future of genetic science. Imagine standing on the edge of the world, looking out over vast horizons that stretch far beyond what the eye can see. From the bustling cities to the quiet fields, the world is full of stories, each one unique. yet all connected by the thread of humanity. As we journey together, we take a soft, reflective look at how different cultures and nations are embracing CRISPR, the groundbreaking gene editing technology that has the potential to change the very fabric of life itself. Each place we visit holds its own perspective on the power of CRISPR and the ethical and scientific questions it raises. In the United States, for example, CRISPR has been greeted with a mixture of enthusiasm and caution. Scientists and researchers are pushing the boundaries of what is possible, working to find cures for genetic diseases like sickle cell anemia and cystic fibrosis. Yet, there is also a strong focus on the ethical questions that arise when gene editing is applied to humans. In the wake of the first human trials, the conversation in America often centers around regulation, safety, and the potential long-term consequences of editing human DNA. The possibility of enhancing human traits, not just curing diseases, has sparked debate. Should we edit genes to enhance intelligence or physical ability? Where do we draw the line between healing and creating designer babies? These questions linger, like a soft whisper, urging us to reflect on what it truly means to shape the human genome. Now, imagine traveling across the ocean to China, where CRISPR technology has also made significant strides. The landscape here is different, both in terms of scientific advancement and the approach to regulation. China has been a pioneer in using CRISPR for research purposes, including editing human embryos. While the potential for breakthroughs in medicine is undeniable, this has sparked significant ethical debates around the world. The Chinese approach to gene editing has often been more permissive, pushing the boundaries of what is considered acceptable in other countries. In some ways, this represents the tension between scientific progress and ethical considerations. Here, as in the United States, the question remains, how do we balance the potential for healing with the responsibility of wielding such powerful technology? From China, we travel to Europe, where CRISPR's use is met with more caution. In countries like Germany, strict regulations govern the use of genetic technologies, reflecting a deep concern for the ethical implications of gene editing. European countries tend to be more conservative when it comes to editing the human genome, particularly when it involves embryos or germline editing, changes that can be passed down through generations. In the European Union, the focus is often on the long-term impacts on society and the environment. The conversation here is deeply rooted in human rights, environmental protection, and the preservation of biodiversity. The European perspective is one of careful deliberation, seeking to avoid the potential for misuse and unintended consequences, while still recognizing the potential of CRISPR to address serious health challenges. As we move south to Africa, we find that CRISPR's potential is seen in a different light. In many African countries, where resources for traditional medicine and healthcare are scarce, CRISPR offers a hope for overcoming diseases that have long been out of reach. Malaria, for example, remains a major problem in sub-Saharan Africa, and scientists are exploring the possibility of using CRISPR to create genetically modified mosquitoes that could stop the transmission of the disease. In this context, CRISPR is seen as a potential tool for addressing some of the continent's most pressing health challenges. But, just like in other parts of the world, there is an underlying question of equity. Who will have access to these life-saving technologies? How can we ensure that CRISPR is used in ways that benefit the most vulnerable populations, without creating new forms of inequality? In South America, the use of CRISPR technology is also being explored, particularly in agriculture. With vast expanses of fertile land and a growing population to feed, CRISPR offers the promise of more resilient crops that can thrive in changing climates. Countries like Brazil and Argentina are at the forefront of using gene editing to create crops that are resistant to pests, diseases, and extreme weather conditions. This technology has the potential to reduce the use of harmful pesticides and increase food security. But as in other parts of the world, the question arises, how do we ensure that these advancements are made responsibly, with an awareness of their impact on the environment and the people who depend on the land for their livelihoods? As we travel the globe, from North America to Asia, from Europe to Africa, and across the Americas, one thing becomes clear: CRISPR is not just a scientific breakthrough, it is a cultural and ethical journey. Each country and region brings its own perspective to the table, shaped by history, values, and priorities. In some places, the focus is on curing diseases and improving human health, while in others, the focus is on the potential for enhancing human traits or protecting the environment. What unites these diverse perspectives is the shared belief in the power of CRISPR to transform lives. But it also highlights the need for a global conversation about the responsible use of this technology, one that takes into account not only the scientific possibilities but the ethical and societal implications. The story of CRISPR is still being written, and it is being written by people from all corners of the world. As the technology evolves, so too will the conversations that surround it. The path forward is one of collaboration, dialogue, and careful consideration. The global landscape of CRISPR is one of vast potential, but also profound responsibility. It is a journey that must be taken together, with respect for the diverse cultures, values, and ethical frameworks that shape our collective future. As you consider this global perspective, the question remains, how will we navigate the challenges and opportunities of CRISPR, ensuring that this powerful tool is used to benefit all of humanity, no matter where they are in the world? And as these reflections settle, gently, a soft thought stirs. What does it mean to approach CRISPR as a global community? And how do we ensure that the promises of this technology are shared equitably across all cultures and nations? I understand your request to strictly adhere to the given word count, and I will ensure that the chapter meets the 1,150 words requirement. Here is the expanded version. Chapter 19. Legal and Regulatory Challenges. What are the boundaries? Imagine the gentle rhythm of a lawmaker's pen, drawing boundaries for the power of CRISPR. This chapter explores the legal and regulatory challenges surrounding gene editing, reflecting on how the law seeks to keep pace with scientific progress. As the sun sets and the world grows quiet, the rhythm of progress continues. In laboratories, in hospitals, in fields, the promise of CRISPR unfolds a tool of great potential, one that can alter the very essence of life itself. With every new discovery, with every experiment, there is a gentle tug of a deeper question. How do we ensure that this power is used responsibly? As we stand on the edge of such profound scientific advances, we must consider how the world will regulate and govern the use of gene editing technology. Just as a gardener shapes the growth of a plant, laws and regulations shape the direction of scientific innovation, providing boundaries to guide its course. Gene editing, with its ability to reshape the genetic code of living organisms, offers a horizon of possibilities. It promises cures for diseases, solutions for food security, and new approaches to human enhancement. Yet, as these possibilities unfold, they raise questions that cannot be answered by science alone. Questions about ethics, about human rights, and about what it means to play God. This is where the law comes in, seeking to provide a framework that ensures CRISPR is used in ways that benefit society as a whole, while protecting the rights and dignity of individuals. The law must balance the potential for innovation with the need for careful consideration, and it must do so in a way that keeps pace with rapidly advancing technology. Imagine, for a moment, the delicate dance between science and law. On one side, there is the constant march of scientific discovery, pushing the boundaries of what is possible. On the other side, there is the slow, thoughtful process of legislation, striving to anticipate and regulate the impact of these discoveries. It is a process that requires patience, understanding, and foresight, for the stakes are high. If we do not set the right boundaries, CRISPR's potential could lead to unintended consequences, including the possibility of misuse. What happens when genetic editing is applied not for healing, but for enhancement? When is it right to intervene in the genetic makeup of future generations? And how do we ensure that these interventions are accessible and ethical, not just in a few privileged parts of the world, but for all of humanity? In the United States, for example, the legal landscape surrounding CRISPR is still being shaped. The federal government has set clear regulations for the use of gene editing in humans, particularly in the context of germline editing, changes made to the DNA of embryos, which can be passed down to future generations, while the use of CRISPR in clinical trials is allowed, especially for treating genetic diseases. There are strict guidelines to ensure that the technology is not used recklessly. In recent years, the idea of designer babies where genes could be edited to select for traits such as intelligence or physical ability has sparked significant debate. Lawmakers are cautious, knowing that while gene editing offers the promise of healing, it also carries the risk of opening Pandora's box, unleashing new forms of inequality, discrimination, and unintended societal consequences. On the other side of the world, in China, the pace of gene editing has been more permissive. Chinese scientists have made significant strides in gene editing, including the controversial creation of genetically edited embryos. In some ways, China has taken a more open approach to CRISPR research, while still grappling with ethical questions. The pace of regulation in China lags behind the rapid scientific advancements, leading to global conversations about the need for international agreements to guide the ethical use of gene editing technologies. The challenge here is clear. How do we harmonize the potential of CRISPR with global ethical standards, ensuring that countries, both developed and developing, use the technology in ways that align with universally accepted principles? In Europe, the legal framework surrounding CRISPR is generally more cautious. The European Union has adopted strict regulations on genetic research, especially when it comes to human gene editing. While gene editing for research purposes is widely accepted, many European countries have placed significant restrictions on editing the human germline. The emphasis is on protecting human dignity, preventing genetic discrimination, and ensuring that gene editing is used only for therapeutic purposes. This caution stems from the lessons of history, where unchecked scientific experimentation has led to ethical and moral missteps. The European approach is a reminder that scientific progress should not come at the expense of humanity's values. As we reflect on the global landscape, it becomes clear that the legal challenges surrounding CRISPR are both complex and urgent. The power of gene editing raises profound questions about our ability to shape life itself, just as science must ensure that it does not overstep its bounds. The law must guide this powerful tool, setting boundaries that allow for innovation, while safeguarding the rights and dignity of individuals and society as a whole. These laws must evolve alongside scientific progress, ensuring that the benefits of CRISPR are accessible to all, not just to those with the means to access cutting-edge technology. They must also address the ethical challenges posed by the technology, ensuring that we do not lose sight of the inherent value of human life and the diversity that makes us who we are. As the debate continues, the future of CRISPR remains uncertain. Will we embrace the full potential of gene editing? And if so, how will we ensure that it is used responsibly? Will we strike the right balance between innovation and ethical considerations, allowing CRISPR to help heal and enhance lives without crossing boundaries we may later regret? The answers to these questions will shape the future of science, law, and humanity itself. For now, the challenge is to ensure that the rhythm of progress is tempered by thoughtful consideration and careful regulation. In the quiet of this reflection, you may feel the weight of these questions settle in, as though the rhythm of progress has slowed, giving time to consider where we go from here. How do we build the legal and ethical framework that will guide CRISPR into a future where it benefits all, without compromising our humanity? And as these thoughts linger softly in the quiet of your mind, a gentle curiosity stirs. What will the future of CRISPR look like, when the laws that govern it are not just created, but carefully woven into the fabric of human ethics and progress? This version is carefully expanded to meet the word count requirement of 1,150 words. Let me know if you would like any further adjustments. Chapter 20. The Cost of Progress. Is CRISPR accessible to all? As we consider the future, we pause to reflect on accessibility. Who gets to benefit from the promise of CRISPR? This chapter gently contemplates the financial and social aspects of gene editing, pondering whether this new technology will be available to all. Imagine, for a moment, a world where the promise of CRISPR is fully realized. Imagine a future where diseases are no longer inherited, where the potential for human health and longevity is greater than ever before. The world is alive with possibility, but as we stand at the threshold of this new era, a quiet question stirs. Who gets to benefit from this promise? Gene editing, with its immense potential, is not just a scientific breakthrough. It is a reflection of the world we want to create. Will the benefits of CRISPR be shared equally, or will they remain locked behind a barrier of privilege? As you consider this, think about the cost of progress. Just as the road to innovation is often paved with challenges and sacrifices, so too is the accessibility of groundbreaking technologies like CRISPR. Imagine, for a moment, that CRISPR is a garden, a field of potential waiting to be cultivated. Some will have the means to reach in and pluck the fruits of this garden, the wealthy, the powerful, those who can afford cutting-edge treatments and enhancements. But for others, this garden may remain out of reach, a distant dream that never touches their lives. In the world of gene editing, the cost is not just measured in dollars, but in access, equity, and opportunity. CRISPR, as we have seen, holds the power to cure genetic diseases, to create stronger crops, and even to enhance human traits. But the question arises, how much will it cost to bring these benefits to the world? The financial barriers are real. For gene editing to become widely accessible, it must be affordable. Yet, the technology is still in its infancy, and the costs associated with developing and applying CRISPR are significant. At this point, the technology is mostly available in well-funded research labs and institutions where the financial backing is strong. Treatments involving gene editing, like the experimental use of CRISPR for genetic disorders, often come with a hefty price tag. Will these treatments remain the domain of the wealthy, or will they one day be available to everyone? Consider the cost of a life-changing treatment. For those suffering from genetic diseases like sickle cell anemia or cystic fibrosis, CRISPR represents hope, a potential cure that could dramatically improve their quality of life. Yet, the price of such hope is often too high for many to bear. In developed countries, where healthcare systems are more robust, access to CRISPR-based treatments may eventually become a reality, but only for those who can afford it. The wealthy may have the privilege of life-saving therapies, while the most vulnerable populations may be left behind. In regions with fewer resources, the challenge of accessing CRISPR may seem even more insurmountable. The promise of gene editing can easily become a distant dream, unattainable for those who need it most. Yet, beyond the financial costs, there are also social and ethical barriers to consider. The availability of CRISPR's treatments is also tied to social and cultural factors. In some societies, the value placed on science and medicine may be stronger, driving investment in gene editing technologies. In others, cultural attitudes towards genetic modification may lead to resistance, or even rejection of CRISPR-based treatments. These social factors play a critical role in determining who benefits from scientific advances. The power to access life-saving therapies will not only be shaped by financial resources, but by the values of each community and society. As we move forward, it is essential that we ask ourselves, how do we ensure that CRISPR's benefits are shared fairly? How can we make sure that this revolutionary technology is accessible to all, not just those with the means to pay for it? It's not just about making CRISPR affordable. It's about making it equitable. This means addressing the broader social and economic factors that influence access to healthcare and technology. It means ensuring that CRISPR is not a tool that deepens the divide between the rich and the poor, but one that helps bridge that gap. One possibility is to focus on creating affordable, accessible treatments that can be distributed more widely. As the cost of CRISPR technology decreases with further advancements in scaling, there may be opportunities to bring it to underserved populations. Public health initiatives could help make gene editing available to those who need it most, without having to rely on the wealth of an individual. In this vision, CRISPR becomes a tool of equity, not just privilege. It becomes a technology that benefits the collective well-being of humanity, not just a few who can afford it. At the same time, it's important to remember that accessibility is not just about price, it's also about infrastructure. In many parts of the world, access to the basic healthcare infrastructure needed to implement gene editing is lacking. Even if CRISPR becomes affordable, it may still be out of reach for people who do not have access to hospitals, clinics, or skilled professionals trained in gene editing. In such regions, the promise of CRISPR may seem distant, a future that is beyond grasp. To make CRISPR accessible to all, we must not only reduce the cost of the technology, but also build the infrastructure necessary to deliver it to every corner of the world. As we reflect on the possibilities, we are reminded that technology alone cannot solve the challenges we face. The true promise of CRISPR lies not just in its ability to cure diseases or enhance human traits, but in how we ensure that these benefits are shared equitably. It is a reminder that science, when combined with compassion and fairness, has the power to create a better world for all. The journey towards a future where CRISPR is accessible to all requires a careful balance between progress and equity, between innovation and justice. The promise of CRISPR can reshape the future of medicine, agriculture, and human health. Yet, as we move forward, we must ask ourselves how we will ensure that this promise is available to everyone. How will we bridge the gap between the technological advances we are making and the realities of access in the world today? As the questions linger, you are gently left with this thought. What kind of future do we want to create with CRISPR? And how can we ensure that it benefits all of humanity, not just those with the resources to access it? Chapter 21. The Ethical Lines. Should we edit the human germline? Should we edit the genes of unborn children? This chapter takes a thoughtful look at the ethical dilemma of germline editing, changing the DNA passed down from one generation to the next. We'll explore the quiet debate that surrounds this powerful capability. As you settle into the quiet of the night, the stars above twinkle softly, their light timeless and far-reaching. In the vastness of the universe, there is a quiet thought that stirs a question, not of the heavens, but of humanity itself. Should we edit the genes of unborn children? Should we take the power of CRISPR and use it to alter the very essence of human life, not just for an individual, but for generations to come? This is the ethical dilemma of germline editing, a tool so powerful that it can change the DNA, passed down from one generation to the next. The implications are profound, and the debate is both quiet and fierce. As we stand on the precipice of a new era, in which the very course of human evolution could be shaped by our hands, imagine, for a moment, the quiet weight of this decision. The possibility of gene editing offers hope, especially for those who suffer from inherited diseases that have plagued their families for generations. What if CRISPR could cure diseases like cystic fibrosis, Huntington's disease, or Duchenne muscular dystrophy before they even have a chance to manifest? The ability to prevent suffering before it begins seems almost like a gift. A promise of a future where no child has to carry the burden of genetic disorders passed down from their parents. Yet, as with all great promises, there is a question that lingers. Should we intervene in such a fundamental way? Should we alter the very building blocks of life, choosing which traits should be passed down and which should not? The ethical debate surrounding germline editing is a reflection of the deeper questions we face as a species. It is a conversation not just about science, but about what it means to be human. Are we playing with fire, meddling with the forces that have guided evolution for millions of years? Or are we, in fact, fulfilling a duty to heal, to improve, and to protect future generations from suffering? As you reflect on these questions, think of the unborn child, waiting to come into the world. What if, through the power of CRISPR, we could ensure that they would be free from the genetic conditions that once threatened their ancestors? What if, by editing their genes, we could give them a healthier life, one without the shadow of disease hanging over them? The potential is extraordinary, but it is also fraught with risks. The decision to alter the genetic makeup of a child is not one to be taken lightly. It is not just about eliminating diseases, but about choosing the very nature of life itself. What does it mean to shape the future of a person before they are born? How do we ensure that we are not creating a world where perfection is the goal and imperfection is cast aside? At the heart of the debate lies the question of consent. How can an unborn child consent to such a profound change in their genetic makeup? What if, in our desire to improve the human condition, we inadvertently create a world where genetic differences are no longer valued, where those who are genetically enhanced are seen as superior to those who are not? The quiet fear that lurks in the debate is the possibility of genetic inequality, a world where the ability to edit genes becomes a privilege for the wealthy, leaving others behind. Could this technology, meant to cure and enhance, instead widen the gap between the haves and the have-nots? As we navigate this question, we must also consider the potential for unintended consequences. The human genome is incredibly complex, and even a small change in one part of it can have ripple effects throughout the entire system. What if, in our desire to correct one genetic flaw, we inadvertently introduce new problems? What if we alter the very essence of what makes us human, the diversity, the imperfections, the very traits that have allowed us to adapt and thrive in a changing world? These are not just scientific questions, they are moral ones, ones that ask us to reflect on the nature of humanity itself. Are we prepared to take responsibility for the long-term effects of altering the genetic code? The ethics of germline editing also raise questions about the value of human life. In a world where genetic conditions can be prevented, would we still value the unique experiences, the individuality, that come with the imperfections of the human condition? If we begin to eliminate genetic diseases, would we also eliminate the struggles that have shaped human resilience and compassion? There is something deeply human about the imperfection of life, about the challenges we face and the ways in which we rise to meet them. As we move toward a future where genetic editing is possible, we must ask ourselves, what are we willing to lose in the pursuit of perfection? The question of germline editing also touches on the idea of control. Who should have the power to decide what changes are made to the human genome? Should the decision be left to scientists, to lawmakers, or to society as a whole? How do we ensure that genetic editing is used responsibly and in ways that benefit all of humanity, not just a select few? These questions are not easily answered, for they require us to confront the very nature of power and responsibility. The ability to shape life itself is a tremendous gift, but it is also a burden, one that must be carried with wisdom and care. As we step back from this reflection, the debate continues. The promise of CRISPR is profound, offering the possibility of a future free from genetic diseases, a future where human suffering can be reduced and health can be enhanced. But with this promise comes the question, where do we draw the line? When it comes to editing the human germline, how do we ensure that we are not crossing ethical boundaries that could forever alter the fabric of humanity? In the quiet of the night, as you reflect on the vast possibilities of CRISPR, the question persists, are we ready to make these decisions? And if so, how do we do so with a deep respect for the human experience? And as these thoughts linger softly, the next question arises. What does it mean to balance scientific progress with the need for ethical responsibility? And how will we guide the future of gene editing in a way that honors both innovation and humanity? Chapter 22. CRISPR and the Rise of Personalized Medicine. Imagine a world where medicine is tailored just for you, based on your unique genetic code. This chapter explores the rise of personalized medicine, where CRISPR plays a key role in creating treatments that are as unique as the individuals they heal. As the world around you gently slows, a quiet thought settles in your mind. What if the treatments we rely on for our health were not only effective, but tailored to who we truly are, down to the very essence of our being? Imagine a world where medicine is designed not just for the average patient, but for you, for me, for everyone, based on our unique genetic code. This world, shaped by CRISPR and the emerging field of personalized medicine, is not as distant as it once seemed. It is a future where the age-old question of how to heal the body becomes more precise, more attuned to our individual needs. In this world, medicine is no longer one size fits all. Instead, it is a deeply personal experience. Your genes, the intricate, unique blueprint that defines who you are, hold the key to unlocking the most effective treatments for your health. What if doctors could analyze your genetic makeup and craft a treatment plan that addresses not only your symptoms, but also the underlying causes of your health issues? This is the promise of personalized medicine, and CRISPR, with its ability to edit and modify genes, is at the forefront of this revolution. Imagine, for a moment, being able to sit down with your doctor and discuss your treatment options. Not in terms of trial and error, but with a deep understanding of your genetic code. Every detail of your genetic makeup would be considered, and, based on that, the treatment you receive would be as precise as possible. If you suffer from a genetic disorder, such as sickle cell anemia, CRISPR could be used to directly alter the faulty gene that causes the disease, offering a cure that is tailored specifically to your genetic needs. If you are at risk for a certain condition, your doctor could use your genetic information to craft a prevention plan, helping you take proactive steps to protect your health. This is a future where illness is no longer treated with broad strokes, but with the fine touch of precision. CRISPR, as a tool for gene editing, allows doctors and researchers to make the most accurate and personalized interventions. It's as if medicine becomes a bespoke garment, woven from the fabric of your own DNA. No longer do we rely on a generic, one-size-fits-all approach. Instead, treatments are designed to fit the unique contours of your genetic code, offering you a healing path that is truly your own. But what makes personalized medicine with CRISPR So powerful is not just its ability to treat illness, its potential to prevent it. Imagine a world where genetic testing is routine, where your doctor can not only identify diseases that may affect you in the future, but also alter your genes to prevent them from taking hold. With CRISPR, this future is not only possible, but becoming increasingly probable. As our understanding of genetics grows, so too does our ability to foresee and prevent diseases before they have a chance to develop. In this way, CRISPR becomes more than a tool for curing illness. It becomes a way of proactively creating health, preventing problems before they even arise. This world of personalized medicine also promises to change the way we think about aging. What if we could use CRISPR to edit the genes that cause age-related diseases, such as Alzheimer's or heart disease? What if we could slow down the aging process itself by repairing the genetic damage that accumulates over time? This is the future that personalized medicine and CRISPR are shaping, a future where the limitations of age are not so fixed, and where the possibilities for maintaining vitality and health well into old age are greater than ever before. However, the potential of personalized medicine also brings with it significant challenges. One of the most profound questions we face is how to ensure that this new world of personalized care is accessible to all. CRISPR, like any revolutionary technology, has the potential to be expensive. If treatments tailored to your unique genetic code are only available to the wealthy, the promise of personalized medicine could create a new divide, one where only the privileged few benefit from the advancements of science. How do we ensure that these breakthroughs are available to everyone, regardless of their economic background? How do we prevent this powerful technology from becoming another tool of inequality? Another challenge lies in the ethical considerations that accompany the ability to edit the human genome. While CRISPR offers the potential to eliminate genetic diseases, it also opens the door to more controversial applications. Should we use CRISPR to enhance human traits, like intelligence or physical abilities? If so, who decides what traits are desirable? And what impact might this have on our sense of individuality and on the diversity that makes us who we are? These questions are not easily answered, and they require thoughtful deliberation as we move forward into an era where the ability to alter human genes is becoming increasingly accessible. As we explore the landscape of personalized medicine, it is important to remember that this new frontier is not without its complexities. Personalized medicine offers the promise of greater precision, greater care, and greater health. But it also requires us to approach it with caution, responsibility, and a deep respect for the individuals it is meant to serve. As we continue to develop and refine CRISPR, we must be mindful of the ethical and societal implications, ensuring that the benefits of this technology are shared by all, and not just a privileged few. The future of medicine, shaped by CRISPR, is one of immense potential, a future where each treatment is crafted specifically for the person receiving it, offering a path of healing that is as unique as they are. But as we stand on the cusp of this new era, we must ask ourselves, how will we ensure that the promise of personalized medicine is not just a vision for the few, but a reality for the many? As you let these thoughts settle softly into your mind, another question lingers. What will it take to create a world where personalized medicine is accessible to all, and where CRISPR is used responsibly to shape a future of health, equity, and opportunity for every individual? Chapter 23. Rewriting Evolution. The promise and perils of human gene editing. In this chapter, we consider the deep, philosophical implications of rewriting human evolution. What does it mean to have the power to alter the course of humanity? As you drift deeper into thought, we reflect on both the promise and peril of such power. Imagine standing on the edge of a vast, infinite horizon, a point in time where the future of humanity is not set in stone, but is instead something we can shape with our own hands. In this place, science and philosophy meet, each urging us to consider the implications of a new power, one that comes with the ability to rewrite the very course of human evolution. What does it mean to have the power to alter the genetic code that has guided humanity for millennia? And as we stand at this threshold, what responsibility do we bear for the changes we make? Human gene editing, with the power of CRISPR, offers us the ability to do what was once thought impossible, to edit the very building blocks of life. With a single change, we could correct genetic defects, eliminate hereditary diseases, or enhance human capabilities. This promise is profound, and it offers hope to countless individuals suffering from conditions that have been passed down through generations. The potential to eliminate genetic diseases, to reduce suffering, to create healthier generations. These are the promises that gene editing holds. Yet, as you think about this power, there is a quiet whisper of caution. The ability to change the genetic code of humans, to rewrite evolution itself, is a force of profound responsibility. With the potential to heal comes the possibility of unintended consequences. The question is not just what we can do, but what we should do. When we edit the genes of individuals, we are not just affecting their health. We are changing the future of humanity. In doing so, we must ask, how do we ensure that these changes do not come at the cost of the diversity, the uniqueness, and the complexity that makes us human? Imagine a world where the genetic makeup of every person is shaped by choice, where traits are selected to ensure health, intelligence, and physical perfection. It sounds like an ideal world, a world free from the suffering caused by genetic diseases. Yet, as the vision of this world unfolds in your mind, you begin to sense the ethical weight of it. What happens when the line between healing and enhancement becomes blurred? If we can choose to eliminate genetic diseases, could we also begin to choose traits we deem desirable, traits that go beyond the need for health and into the realm of personal preference? Could we create a world where genetic modification becomes the norm, and the very concept of natural diversity begins to fade? The promise of human gene editing lies in its ability to address the challenges of disease and health. But the peril lies in the potential to overreach, to use this technology to enhance human traits, creating a society that values genetic perfection over the imperfections that define our humanity. Consider the possibility that in our pursuit of better health, we might inadvertently create a world that celebrates uniformity rather than individuality. The beauty of humanity, after all, lies in its diversity, in the range of experiences, backgrounds, and perspectives that shape us. If we begin to edit out what we perceive as flaws, might we also lose something irreplaceable about the human spirit? As you reflect on these possibilities, the question deepens. Who gets to decide which traits are worth enhancing? If gene editing becomes a tool for creating perfect individuals, who sets the standards for what is considered perfect? This question touches on issues of power and control. The ability to edit human genes carries with it the potential for misuse. In a world where genetic modification is possible, those who control the technology could gain unprecedented power to shape not just the future of individuals, but the future of entire populations. How do we ensure that these technologies are used in ways that are ethical, just, and equitable, rather than reinforcing existing inequalities? Another question that arises as we consider the implications of rewriting evolution is one of consent. If we edit the genes of unborn children, those individuals have no say in the decisions that shape their genetic makeup. What does this mean for personal autonomy? Should we have the right to shape the genetic future of our children, even if it means making choices for them before they are born? The power to edit human genes raises fundamental questions about the nature of freedom, consent, and individual rights. How do we navigate this power with respect for the autonomy of future generations? Despite the concerns, The promise of gene editing is undeniable. The potential to eradicate diseases, to prevent suffering, and to offer new opportunities for health and longevity is a gift that could change the course of humanity. However, as we stand at the crossroads of this new era, we must proceed with care. The power to rewrite evolution is a responsibility that requires thoughtfulness, humility, and deep consideration of the consequences, both seen and unseen, of the choices we make. We must ask ourselves not just what is possible, but what is right. What kind of world do we want to create with this power? And how do we ensure that the future we shape is one that reflects our shared values and respect for the diversity of life? As we explore the future of human gene editing, we are reminded that science is not just a tool for progress, it is a tool for understanding. With the ability to change the human genome comes the responsibility to reflect on what it means to be human. Our genetic makeup is not just a collection of instructions. It is a reflection of our history, our struggles, and our triumphs. In editing the genes of future generations, we must not lose sight of the very essence of what makes us who we are, our humanity, our imperfections, and our shared experience. As you consider these reflections, you may feel the weight of the questions we face. But within this uncertainty lies the opportunity for thoughtful dialogue and responsible action. The promise of gene editing is a gift, but it is one that must be handled with care. As we move forward, we must ask ourselves, how do we balance the promise of progress with the responsibility of preserving the diversity, complexity, and beauty of the human experience? And as these reflections settle, a quiet curiosity arises. What ethical boundaries will we establish as we move toward a future where human gene editing is not just possible, but a reality? Chapter 24. A New Era of Medicine, CRISPR, and Cancer. Treatment. In the quiet moments before sleep, we consider the possibilities CRISPR holds for fighting cancer. Imagine using gene editing to target cancer cells with the precision of a surgeon's scalpel. This chapter explores how CRISPR is being used in cancer research and treatment. As the world around you quiets and the weight of the day fades, your mind drifts to the vast expanse of possibilities that lie ahead. There, just beneath the surface of sleep, there is a new world of healing. One where science has unlocked a precision so delicate that it can cut through the very fabric of life itself. This world, shaped by CRISPR, brings with it the hope of a new era in medicine, a future where diseases that once seemed insurmountable, like cancer, could be met with a weapon as precise as a surgeon's scalpel. Imagine, for a moment, a world where the very cells that threaten to destroy the body can be singled out and corrected before they can do their damage. Cancer, in all its forms, remains one of the most formidable challenges facing medicine today. It is a disease born of our own cells, one in which healthy cells begin to lose their way, growing uncontrollably and forming tumors. Despite decades of research, treatments have often been blunt instruments: chemotherapy, radiation, and surgery, each with its own set of risks and side effects. Yet, as we stand on the threshold of this new era, CRISPR offers a different way, one that promises to treat cancer with a level of precision that was once unimaginable. Imagine a future where CRISPR is used to edit the genes of cancer cells themselves, to directly target the mutations that cause them to behave abnormally, instead of bombarding the body with generalized treatments that affect both healthy and cancerous cells alike. CRISPR could be used to surgically edit the DNA of the cancer cells, removing the mutations that drive their unchecked growth. This is not science fiction. It is a reality that is slowly unfolding in laboratories around the world. At the heart of CRISPR's potential in cancer treatment is its ability to make precise changes to the genetic code. In cancer, the mutations that cause cells to divide uncontrollably are often the result of errors in the DNA. These mutations can vary widely between individuals and even between different types of cancer. CRISPR offers the ability to target these specific mutations, editing the cancer cell's DNA directly and halting their growth. By cutting out the faulty genetic material and replacing it with healthy, functioning DNA, CRISPR could potentially turn cancerous cells back into normal, healthy ones. This approach, while still in its early stages, is a revolutionary step forward. Imagine, for a moment, that instead of chemotherapy, which destroys both healthy and cancerous cells, you receive a treatment that seeks out only the cancer cells and alters them at their genetic level. With CRISPR, it is possible to imagine a world where cancer is treated not by simply attacking everything in its path, but by surgically removing the disease at its source, the mutated genes that drive it. This would not only increase the precision of treatment, but also minimize the side effects that so many patients endure during traditional therapies. But there is more. CRISPR's potential in cancer treatment goes beyond just editing the genes of cancer cells. Researchers are exploring the possibility of using CRISPR to modify the immune system itself, making it more capable of recognizing and attacking cancer cells. In this scenario, CRISPR could be used to teach the immune system to better identify cancer cells, turning the body's natural defenses into a more effective weapon against the disease. This is known as immunotherapy, and CRISPR's ability to enhance the immune system could make it an even more powerful tool in the fight against cancer. Imagine that your body's immune cells are like tiny soldiers, patrolling your body and defending it from invaders. In the case of cancer, however, these soldiers often fail to recognize the cancer cells as a threat. CRISPR offers the possibility of editing immune cells so that they can more effectively identify and destroy cancer cells. This personalized approach would not only be more precise, but would also reduce the collateral damage that often occurs with traditional treatments. The potential of CRISPR to target cancer so precisely is both exciting and humbling. However, as with any powerful tool, the path forward is not without challenges. Cancer is a complex, ever-changing disease, and the mutations that drive it can vary greatly between individuals and even within different areas of a single tumor. This means that CRISPR's ability to target cancer cells will need to be as adaptable and varied as the disease itself. Researchers are still working to understand how to best use CRISPR to target cancer cells while avoiding potential side effects or damage to healthy cells. Precision is key, and while CRISPR offers incredible promise, it must be used with caution and care. Another challenge lies in the delivery of CRISPR to the right place in the body. Gene editing, by its very nature, involves changing the DNA within cells, and getting CRISPR into those cells, particularly cancer cells, is no small feat. Scientists are working on developing methods to deliver CRISPR more effectively, using nanoparticles or viral vectors to carry the gene editing tools to the right locations. It is a delicate process, requiring both precision and innovation to ensure that the treatment reaches the cancer cells without causing harm elsewhere in the body. But despite these challenges, the potential is undeniable. CRISPR represents a new frontier in cancer treatment, one where precision and personalization are at the heart of the approach. Rather than treating cancer with a one-size-fits-all method, CRISPR offers the possibility of tailoring treatments to each individual, targeting the very genetic mutations that drive their disease. It is a future where cancer treatments are not just more effective, but also more compassionate, focused on healing the body with the gentleness and precision of a skilled surgeon. As we reflect on the promise of CRISPR in the fight against cancer, we are reminded that this new era of medicine is still unfolding. The road ahead is one of discovery, experimentation, and careful testing. But with every step forward, the hope grows stronger that one day, cancer may no longer be the silent, unstoppable force it once was, but a disease that can be defeated with the precision and grace of gene editing, in the stillness of the night. As these possibilities gently settle into your thoughts, you are left with one quiet, persistent question. How will we navigate the future of CRISPR in cancer treatment, ensuring that its power is harnessed with care and used for the healing of all? And as you ponder the potential of CRISPR, another thought arises. What role will personalization play in the future of medicine, and how will it reshape not just cancer treatment, but our entire approach to health and healing? Chapter 25. The emotional impact of genetic engineering. What does it mean to be human? As we explore the emotional landscape of genetic engineering, we pause to consider what it means to be human. Does editing our genes change who we are? This chapter delves into the psychological and emotional implications of gene editing, pondering the essence of our humanity in the stillness of the night. As you drift between wakefulness and sleep, a thought quietly emerges. What does it mean to be human? It's a question that has been asked for centuries, one that touches on our deepest fears, hopes, and aspirations. Now, in this new age of genetic engineering, this question takes on new dimensions, with the ability to alter our genetic makeup, to change the very essence of our biology. The line between what is natural and what is created begins to blur. As you think about this, you wonder, does changing our genes change who we are? Imagine, for a moment, that the blueprint of your very being, your genes, could be rewritten. CRISPR, the tool that allows for such precise edits, offers us the possibility of erasing genetic diseases, of enhancing human traits, and even of crafting future generations with specific attributes. It's a future that promises health, vitality, and progress, but also one that raises emotional questions about identity, choice, and the very fabric of our humanity. If we can change the code that makes us who we are, does that alter the essence of our being? As you reflect on this, the weight of the question deepens. What happens to the part of us that is shaped by our genetic inheritance? Our genes are not just a collection of instructions. They are a part of our story, passed down through generations. They are the markers of our ancestors, the remnants of their struggles, triumphs, and unique experiences. To edit our genes, to change the course of our genetic history, is to alter that story. Does that make us less connected to the past? Does it make us less human? The emotional implications of genetic engineering are profound. Consider, for example, the possibility of eradicating genetic diseases that have plagued families for generations. In the past, these diseases were a heavy burden, passed down from one generation to the next. With CRISPR, there is a chance to lift this burden, to erase the genetic flaws that cause suffering. On the surface, this seems like an unequivocal good, a way to free future generations from the pain and hardship of inherited diseases. Yet, as we imagine this future, we must ask ourselves, what does it mean to live in a world without these struggles? Is part of what makes us human our ability to overcome adversity, to learn from hardship? And if we remove the potential for these challenges, do we lose something essential in the process? Similarly, the idea of enhancing human traits raises deeper emotional questions. If we could use CRISPR to enhance intelligence, strength, or even emotional resilience, what would it mean to live in a world where such traits are no longer left to chance? The idea of designer babies, children whose genetic traits are selected before birth, stirs up powerful emotions. On one hand, it offers the possibility of creating individuals who are healthier, stronger, and more capable of navigating the world. On the other hand, it raises the question of what it means to be truly unique. If we can choose the traits we value most, do we lose the beauty of human diversity, the imperfections that make us who we are? In the quiet reflection that follows, you may begin to wonder, what is the true value of human diversity? Our differences in appearance, temperament, and even abilities, are what make us unique, and yet, gene editing presents the possibility of removing those differences in the pursuit of perfection. If we shape the future of humanity to fit a narrow standard of beauty, intelligence, or physical ability, do we risk losing the richness of the human experience? The imperfections that define us as individuals are often the very things that give us depth, empathy, and resilience. In striving for a world free of disease and suffering, do we risk creating a society where only certain traits are valued, and all else is seen as undesirable? The emotional impact of genetic engineering also touches on the very concept of identity. Who are we if our genes can be altered, edited, or rewritten? Our genes are not just biological codes. They are part of our personal and familial identity. They connect us to our ancestors, to the generations that have come before us. When we change our genes, we are, in a sense, altering the connection we have to our past, to our lineage. What does it mean to be a person in a world where the very essence of who we are can be modified, customized, or even erased? There is also the matter of consent. If our genetic makeup can be edited before birth, who decides what changes should be made? Can we justify altering the genes of a child who has no say in the matter, simply because it may lead to a better life? The act of altering the human genome raises profound ethical questions about autonomy, choice, and the right to shape one's own future. The emotional weight of these questions is heavy, as they force us to confront the boundaries between science, ethics, and human dignity. As you consider these emotional landscapes, you may feel a quiet sense of wonder. The promise of CRISPR and genetic engineering is vast. It offers the potential to end suffering, to create healthier individuals, and to enhance human capabilities in ways we have never dreamed possible. Yet, the journey ahead is not just one of scientific discovery, but of deep introspection and careful reflection. As we venture into this new world, we must consider not just the technical possibilities, but the emotional and philosophical implications of altering the very essence of what it means to be human in the stillness of the night. As you drift deeper into thought, the question lingers: what does it mean to be human in a world where we have the power to change our very DNA? Is our humanity defined by our genetic code, or by the experiences, struggles, and connections that shape us? And as we stand on the threshold of this new era, we must ask ourselves, how do we navigate the emotional and ethical complexities of genetic engineering, ensuring that we preserve the essence of our humanity while embracing the possibilities of this powerful technology? And as these reflections settle gently in your mind, a soft question stirs. How will we reconcile the promise of genetic engineering with the emotional and philosophical questions it raises about identity, diversity, and the very nature of being human? Chapter 26. CRISPR's Place in the Future of Medicine. A silent revolution. In this chapter, we quietly reflect on the revolution that is happening behind the scenes in medical labs. CRISPR is not just a tool, it is a silent revolution. We'll explore how its widespread use could change the future of medicine forever. As the evening settles around you, a soft calm begins to wash over your mind. The world outside grows quiet, and in this stillness, you reflect on the gentle but profound shifts that are occurring in the world of science and medicine. Behind the closed doors of laboratories and research centers, a revolution is quietly unfolding. It is not a loud, dramatic upheaval, but a subtle, almost unnoticed change, one that is shaping the future of medicine in ways we are only beginning to understand. This revolution is the advent of CRISPR, a tool so powerful, yet so precise, that it has the potential to reshape the way we approach the healing of the human body. CRISPR, in its simplest form, is a tool for editing genes, the very building blocks of life. With the ability to cut, modify, and replace strands of DNA, CRISPR holds the key to a future where diseases that once seemed untouchable can be treated at their very source. Imagine a world where genetic disorders like sickle cell anemia, cystic fibrosis, and even certain cancers can be cured not by treating symptoms, but by correcting the underlying genetic mutations that cause them. This, in essence, is the silent revolution that is taking place, a quiet force that promises to transform the landscape of medicine forever. But this revolution is happening slowly, behind the scenes, away from the public spotlight. While headlines may focus on the dramatic breakthroughs in CRISPR research, much of the progress is being made in small, steady steps. As scientists work tirelessly in labs around the world, these incremental advancements are building the foundation for a future where gene editing is not just a possibility, but a reality, one that will impact everything from genetic disease treatment to the very way we approach human health. Imagine, for a moment, a world where medical treatments are as tailored as a perfectly fitted garment, designed specifically for you. With CRISPR, that future becomes more plausible. The ability to edit genes opens the door to personalized medicine, a world where treatments are crafted not based on averages, but based on your unique genetic code. This could mean that, rather than guessing at what treatment will work for you, doctors could analyze your DNA and design a therapy that targets your specific genetic makeup. It's a world where medicine is no longer one size fits all, but one where every treatment is as unique as the person receiving it. The potential for CRISPR in treating diseases is vast. Take cancer, for example. Traditional treatments like chemotherapy and radiation often target cancerous cells, but they also harm healthy cells along the way, leading to unwanted side effects. CRISPR, on the other hand, offers the possibility of directly targeting the genetic mutations that cause cancer. Rather than bombarding the entire body, CRISPR could selectively edit the genes in cancer cells, removing the mutations that allow them to grow uncontrollably. This approach, when fully realized, could revolutionize cancer treatment, offering a way to heal the body with unprecedented precision. Yet, this revolution is not without its challenges. While the promise of CRISPR is undeniable, there are still hurdles to overcome. The technology, though powerful, is still in its infancy. The ability to accurately and safely edit genes within the human body requires an understanding of genetics that is still evolving. Scientists are working to refine the delivery methods, ensuring that CRISPR reaches the right cells without causing unintended effects. They are also exploring ways to minimize the risk of off-target edits, where CRISPR might make changes to genes that were not intended to be altered. These challenges are part of the quiet revolution, as researchers work steadily toward a future where CRISPR can be used safely and effectively in medical treatments. Even as these challenges remain, the potential of CRISPR is already beginning to manifest in the treatment of genetic diseases. In clinical trials, researchers have used CRISPR to edit the genes of patients with inherited diseases, offering hope where there was once only uncertainty. These early trials are just the beginning, and though the results are still being studied, they mark a turning point in how we view gene editing in medicine. What was once science fiction is slowly becoming science fact, and the world of medicine is beginning to shift in response. The quiet revolution that CRISPR is ushering in goes beyond just curing diseases. It holds the potential to unlock new ways of thinking about health, longevity, and the very nature of the human body. What if we could use CRISPR, not just to heal, but to enhance, to help the body fight diseases before they even begin? What if CRISPR could be used to improve the body's natural defenses against viruses or environmental stressors? The possibilities are vast, and as the technology continues to evolve, so too does the potential for new, life-changing treatments. But as we contemplate the promise of CRISPR, we must also consider its ethical implications, the ability to edit. The human genome is a powerful tool, but it is one that must be used with care. We must ask ourselves, how far should we go in editing the genes of future generations? What happens when the power to edit our genes extends beyond curing diseases and into the realm of enhancement? These questions are not easily answered, and they require us to think deeply about the kind of future we want to create, one that respects the sanctity of human life, while also embracing the potential for progress. The quiet revolution of CRISPR is not just about changing the way we treat diseases. It is about changing the way we think about the human body. It is about reimagining the possibilities of medicine and health, and opening the door to a future where the tools to heal are not just found in pills or procedures, but in the very code that makes us who we are. It is a revolution that is still unfolding, step by careful step, and as we look to the future, we must ask, how will we harness the power of CRISPR, not only to heal, but to ensure that this new era of medicine benefits all of humanity? As these thoughts gently settle, a question arises in the quiet of your mind. What role will CRISPR play in the evolution of medicine? And how will we guide this silent revolution toward a future where it heals, enhances, and respects the essence of life? Chapter 27. Looking to the stars. Can CRISPR help us reach beyond Earth? Imagine using CRISPR to help humanity reach the stars, modifying human genes to withstand the harsh conditions of space. This chapter gently imagines a future where CRISPR could be key to surviving in the cosmos. As you close your eyes and feel the weight of the world slowly ease, a gentle thought arises, one that carries you far beyond the present, toward the endless expanse of the cosmos. In the vastness of space, there is a silence unlike any other, an infinite mystery that stretches out before us. Humanity has always gazed upward, dreaming of the stars, imagining a future where we venture beyond the boundaries of Earth. But the road to the stars is not one without challenges. The journey to space requires more than just technology and innovation. It requires us to adapt, to stretch the very limits of human biology. Could CRISPR, this powerful tool for editing genes, play a role in our ability to survive and thrive in the harsh conditions of space? As you imagine this future, think of the vast challenges that space presents. The vacuum of space, with its extreme temperatures, radiation, and lack of atmosphere, is a far cry from the familiar warmth and protection of Earth's surface. The human body, as resilient and adaptable as it is, was not designed to withstand the rigors of space travel for long-duration missions, such as journeys to Mars or beyond. The effects of space travel on the human body can be profound, from muscle atrophy to bone density loss, from weakened immune systems to changes in the very DNA that governs our cells. These challenges are not just physical, they are biological, deeply embedded in the very fabric of our being. How can we adapt to these new frontiers, where the body's natural limits are tested in ways we've never experienced before? This is where CRISPR enters the picture. Imagine, for a moment, that we could modify the very DNA of astronauts, making their bodies more resilient to the harsh conditions of space. CRISPR could be used to edit genes that govern bone density, muscle strength, and immune function, altering them in ways that allow humans to withstand the physical toll of space travel. Perhaps, in the future, we could edit the genes that make us susceptible to radiation, offering protection against the harmful cosmic rays that flood the vacuum of space. These modifications would not only help us survive in space, but enable us to thrive, ensuring that the human body can endure the challenges of long-duration space missions. In this imagined future, CRISPR could be the key to unlocking human adaptation to space. It could allow us to modify our biology in ways that prepare us for the extreme conditions of other planets, or even the void of space itself. For instance, CRISPR could be used to help astronauts resist the effects of radiation exposure by editing genes involved in DNA repair mechanisms. Space radiation is a constant threat to astronauts, and its damaging effects on human cells could be mitigated by genetic modifications that enhance the body's ability to repair DNA damage. By altering these genetic pathways, CRISPR could help protect the body from the harmful effects of cosmic rays, allowing humans to explore the stars with greater safety and confidence. Similarly, CRISPR could be used to modify the human immune system, preparing it to face the unknown challenges of space. In the sterile environments of spacecraft or colonies on distant planets, the immune system's ability to adapt to new pathogens or conditions is essential. By editing the genes responsible for immune response, scientists could enhance the body's ability to fight off infections, even in environments where Earth-based microbes might not exist. This would allow humans to stay healthy, even as they journey across the cosmos, free from the illnesses that could otherwise derail a mission. But, as you think about these possibilities, a quiet question emerges. While CRISPR offers the potential to adapt humans for space travel, what does this mean for the essence of our humanity? The more we modify our genes to survive in space, the more we begin to change the very nature of what it means to be human. If we can edit genes to adapt to extreme environments, do we risk losing the qualities that make us uniquely suited to life on Earth? The question is not just about survival. It is about what it means to remain human in a future where our biology can be altered at will. This is the paradox at the heart of CRISPR's role in space exploration. The technology that promises to help us reach new frontiers also challenges us to consider the very essence of who we are. If we modify our genes for space, what are we willing to change? Are we willing to sacrifice parts of our biology that we have always taken for granted? What is gained and what is lost when we step beyond Earth and into the stars, armed with the power to reshape our very DNA? In this quiet moment, as you imagine the future of space travel, you are reminded of the vast potential CRISPR holds, not just for curing diseases or enhancing human abilities, but for preparing us for the unimaginable. The dream of reaching the stars is no longer just about rockets and technology. It is about adapting ourselves, through gene editing, to thrive in environments that were once inhospitable to life. With CRISPR, we could rewrite the limits of human biology, opening up possibilities that were once the stuff of science fiction. And yet, as with all powerful technologies, the path forward requires careful thought and reflection. While CRISPR holds the promise of unlocking new frontiers in space, it also requires us to consider the ethical implications of altering human biology on such a grand scale. As we push the boundaries of human exploration, we must ask ourselves, how far should we go in altering our genes? What does it mean to be human when we can change the very building blocks of life itself? These are the questions that will guide us as we move toward a future where the stars are within our reach, but the very nature of humanity itself may be changed forever. As you reflect on these possibilities, you are left with a quiet wonder. What does the future hold for humanity as we prepare to journey to the stars? And how will CRISPR shape not just our biology, but the essence of who we are? And as the stillness of the night wraps around you, a thought stirs. How do we balance the promise of CRISPR in space exploration with the need to preserve the core of human identity as we expand beyond Earth's embrace? Chapter 28. Reflections on Life, Health, and the Future of Genetic Engineering. As we near the end of our journey, we pause to reflect on the delicate balance between life, health, and the future of genetic engineering. This final chapter gently considers the lessons we've learned and the promise of a future where science and ethics walk hand in hand. As the night deepens and the world outside falls quiet, there is a moment of stillness, a gentle pause in time. you find yourself at the end of a long and thoughtful journey, one that has taken you through the realms of science, possibility, and reflection. Throughout our exploration of genetic engineering, from the precise power of CRISPR to the profound ethical questions it raises, we've walked together, guided by the promise of a future where science could heal and transform humanity. Yet, as we near the end of this path, a deeper question lingers, quietly inviting us to reflect. What does it all mean for life, health, and the future of our humanity? Throughout this journey, we have witnessed the incredible potential of genetic engineering. We've seen how CRISPR holds the promise of curing genetic diseases, enhancing health, and even preparing us for the challenges of space. We've imagined a future where diseases like cancer, cystic fibrosis, and muscular dystrophy are no longer a threat, where the very genetic code that defines us can be edited, refined, and improved. The possibilities are immense. As though we've unlocked a door to a new chapter in human evolution, one where the frailty of human health can be mitigated through the careful, deliberate application of genetic knowledge. Yet, as you consider these possibilities, there is a quiet realization that the power to alter life on such a fundamental level requires a delicate balance. To edit the genetic code is to rewrite the story of life itself. Every decision we make in the realm of genetic engineering carries with it the weight of responsibility, and the ethical questions we've faced along the way become more poignant. The promise of healing and enhancing life is undeniable, but how do we ensure that we do so without losing the very essence of what makes us human? It is a delicate dance between progress and caution. As science continues to evolve, we find ourselves at the crossroads of possibility and prudence. We've imagined the ways in which CRISPR can enhance human health, protect us from genetic diseases, and potentially make us more resilient to the harsh realities of space and time. But in all of these promises, there remains a core question. What are the ethical implications of these advancements? The power to edit human genes, to create life with specific traits, carries with it both hope and concern. If we can cure diseases, should we? If we can enhance human abilities, should we? And, perhaps most profoundly, if we can alter the very essence of what it means to be human, should we? Throughout this journey, we have learned that science does not exist in a vacuum. It is deeply intertwined with the moral fabric of society, and the choices we make today will reverberate throughout future generations. Genetic engineering, in its essence, is a mirror of our values and our vision for the future. It is not merely a tool to shape life, but a reflection of who we are as a species, and how we wish to define ourselves in the face of such profound power. As we have seen with CRISPR, there is both great potential and great risk in wielding this tool. In our exploration, we've also touched upon the importance of accessibility and equity. The promise of CRISPR is immense, but it will only truly transform humanity if it is made accessible to all. We cannot afford to create a world where genetic engineering becomes a privilege for the wealthy or a tool for inequality. The future of genetic engineering must be one where every person, regardless of their background or resources, can benefit from the advances in science. It is not just about curing disease for some, but ensuring that all have the opportunity to live healthier, more fulfilling lives. The delicate balance we must strike between innovation and equity is one of the defining challenges of our time. As science pushes forward, we must be ever mindful of the social, cultural, and economic implications of genetic engineering. What will the world look like when gene editing becomes commonplace? Will we value diversity, or will we pursue a vision of perfection? These are questions that require us to not only look forward, but to also reflect on the values that guide us as a society. The promise of genetic engineering is not just in its potential to heal the body, but in its ability to strengthen the very fabric of human connection, to create a world where everyone, regardless of their genetic makeup, has the opportunity to live a full and healthy life. As we consider the future of health and genetic engineering, we must also acknowledge the philosophical questions it raises. What does it mean to be human? How do we define life, health, and well-being in a world where our biology is no longer fixed, but can be shaped by our choices? These questions are not easily answered, but they are essential to the ongoing conversation about the role of science in our lives. The answers we find will shape not only the future of medicine, but the future of humanity itself. The journey we've taken through the world of genetic engineering has been one of awe and contemplation. From the hope of curing diseases to the ethical dilemmas of genetic enhancement, we've explored the vast potential of CRISPR and its role in shaping our future. Yet, as we reach the end of this chapter, we are reminded that the true promise of genetic engineering lies not just in its ability to alter our genes, but in its ability to enhance our humanity. The future we envision must be one where science and ethics walk hand in hand, where the power to heal and enhance life is balanced by a deep respect for the diversity, complexity, and beauty of the human experience. And so, as you drift deeper into thought, the quiet question remains: what kind of world do we want to create, where the promise of genetic engineering is not just a tool for progress, but a way of strengthening our shared humanity? And as the stillness surrounds you, another thought stirs. How do we ensure that the power to shape our future is guided by the values of compassion, fairness, and respect for life as we step into this new era of genetic possibility? Chapter 29. Beyond the Edge. What lies after CRISPR? As the stars fade into the horizon, we look beyond CRISPR. What new technologies will come next? This chapter explores the future of gene editing and what lies beyond the cutting edge of CRISPR. As the soft whispers of night envelop you, a calm reflection arises, a quiet pause, inviting you to look beyond the world we know, to the vast expanse of possibility that stretches before us. We have ventured deep into the world of CRISPR, exploring its power to shape life, heal diseases, and open doors to a future that was once unimaginable. But as the stars flicker in the distance, another question emerges: what lies beyond CRISPR? As the cutting edge of genetic engineering, CRISPR has already made an indelible mark on science and medicine. But like the stars in the night sky, it beckons us toward further exploration into the realms of technologies yet to be discovered. CRISPR, in its brilliance, is a revolution, a tool that has allowed us to edit genes with precision and grace, changing the very fabric of life itself. But as we have seen, even the most groundbreaking technologies have their limits. CRISPR, for all its power, is still a young technology, one that is evolving and growing in the hands of scientists and researchers. As you think about the future, the question becomes not just what is possible with CRISPR, but what lies beyond it. The horizon of genetic engineering stretches far beyond the reach of any single tool, and new technologies are already beginning to emerge, each promising to build upon the foundation that CRISPR has laid. One possibility lies in the realm of base editing, a technology that allows for even more precise changes to the genetic code than CRISPR, whereas CRISPR works by cutting and reassembling the DNA. Base editing can alter individual DNA bases without making cuts. This provides a finer level of precision, offering the potential to correct genetic mutations with even greater accuracy. Base editing, like CRISPR, has the ability to correct disease-causing mutations at their source, but it does so without the risk of unintended changes that could arise from the cutting process. The promise of this technology lies in its ability to make more subtle, targeted alterations to the genome, and it could be the next step in achieving safer and more effective gene editing. As you consider these advancements, the horizon widens further. There are also emerging techniques, like prime editing, that promise to revolutionize the way we approach gene editing. Prime editing, often described as CRISPR 2.0, has the potential to perform more accurate edits by using a guide RNA and an enzyme to insert new genetic material precisely where it is needed. Unlike CRISPR and base editing, which rely on cutting the DNA, prime editing works by rewriting DNA without cutting, offering a level of precision that could change the landscape of gene therapy. With prime editing, we could potentially correct a wider range of genetic disorders, including those that CRISPR currently struggles to address, and with greater efficiency and safety. The future of gene editing, as seen through the lens of prime editing, holds a promise that is both profound and humbling: the ability to repair the very instructions that shape our existence with unprecedented accuracy. But even beyond these technologies, the future of gene editing may include the development of synthetic biology, an emerging field that seeks to not only edit genes, but also design new ones from scratch. Synthetic biology allows us to not just modify existing DNA, but to create entirely new genetic sequences, to write new life forms and organisms that have never existed before. This could open up possibilities far beyond healing genetic diseases. Imagine, for a moment, creating organisms that can help us address the environmental challenges of our time, like microorganisms that can clean up pollution, or plants that can absorb more carbon from the air. Synthetic biology, with its capacity to create new life forms, could become an indispensable tool for reshaping the world, creating a new chapter in our relationship with nature itself. Yet, as with all powerful technologies, these possibilities come with their own set of challenges and ethical questions. With each new advancement, we must ask ourselves, what are the consequences of creating life, altering genomes, or designing new organisms? As we push the boundaries of science, we are faced with the profound responsibility of ensuring that these technologies are used wisely and ethically. It is not enough to simply explore what is possible. We must also consider how these advancements fit into the larger tapestry of humanity, how they affect the world around us, and how they shape the future of life on Earth. The potential to edit and create life is both exhilarating and humbling. With every new step forward, we must tread carefully, guided by both the promise of these technologies and the ethical questions they raise. The ability to manipulate the genetic code is not just a scientific achievement. It is a moral responsibility, one that requires us to ask deep questions about the future we wish to create. Will we use these technologies to heal and enhance, or to control and divide? How can we ensure that the benefits of gene editing and synthetic biology are shared by all, and that the power to shape life is wielded with respect for its complexity and diversity? As you reflect on these possibilities, the horizon before you seems infinite, with the future of genetic engineering expanding into uncharted territories. The promise of CRISPR base editing, prime editing, and synthetic biology holds the potential to transform our world in ways we have only just begun to imagine. But as we look beyond the edge of these technologies, the question becomes not just what will we create, but how will we guide these creations with wisdom and humility? The future of genetic engineering is not just a scientific endeavor. It is a philosophical journey, one that asks us to reconsider what it means to be human, what it means to create life, and how we can ensure that this power is used for the greater good. As you let these reflections settle softly, you may find yourself gently pondering, how will we navigate the uncharted territories beyond CRISPR, ensuring that the advancements we make serve not only science, but humanity itself? As you drift deeper into thought, the quiet question stirs. What is the future we want to build with the tools of genetic engineering? And how can we ensure that we move forward with responsibility, compassion, and respect for the life we seek to shape? Chapter 30. The journey continues. Science and the infinite possibilities of life. In this final meditation, we reflect on the infinite possibilities of life, always changing, always evolving. With CRISPR, we've glimpsed the future, but the journey doesn't end here. This chapter encourages you to dream about what could be, and how science will continue to shape our lives. As the soft glow of the night deepens, you feel a sense of quiet peace wash over you. The journey we've taken together, through the wonders of genetic engineering and the potential of CRISPR, has been profound, yet there is a subtle invitation to look beyond what we've learned, to peer into the vast, uncharted future that lies ahead. For all the advances we've made in science, this is not the end, but a beginning. With each discovery, a new path unfolds, and the possibilities stretch out before us like an endless horizon. In this stillness, we pause to reflect on what could be, on the infinite possibilities of life, a life that is always changing, always evolving, forever reaching toward something new. With CRISPR, we've glimpsed a future where genetic diseases may be erased, where human traits could be enhanced, and where the very building blocks of life are no longer set in stone. Yet even as we stand on the edge of this incredible moment, it is important to remember that the journey does not stop here. The technology we have today is merely the first step, a glimmer of what is to come. As we consider the future, we must remember that science is never static. It is always evolving, always expanding. Just as we look at CRISPR and see the profound ways it is shaping our world, we must also look forward with wonder, dreaming of what the next discoveries will bring. Imagine, for a moment, a world where genetic engineering is commonplace, where treatments and enhancements are as natural as the medicines we take today. What if gene editing could be used not only to cure diseases, but to help us adapt to the challenges of a changing planet? Could we use CRISPR to create crops that thrive in harsher climates, to improve the resilience of ecosystems, or even to help humans adapt to new environments like the Moon or Mars? The future of genetic engineering holds these possibilities, and more, as we continue to unlock the secrets of life at the molecular level. Yet, as you dream about this future, there is a quiet question that remains. What does it mean for us, as human beings, to wield such power? If we can edit our genes, our very essence, what becomes of our natural imperfections, our diversity, and the unpredictability of life? What happens to the delicate balance of nature, the very fabric of what makes us human? With every leap forward in science, we are confronted with deeper questions about our identity, our values, and our role in the world. These questions, like whispers in the night, continue to guide us as we move forward, always aware of the responsibility that comes with such power. The journey we've taken is not just about understanding the mechanics of CRISPR or genetic engineering. It's about contemplating how these tools fit into the broader narrative of life itself. Science, after all, is not just about advancing technology. It is about exploring what it means to live, to thrive, and to be human in a universe that is constantly changing. In a world where the boundaries of life are no longer fixed, where we can shape and reshape the very building blocks of existence, the journey of discovery becomes one of introspection, reflection, and ethical consideration. As you drift deeper into thought, consider the journey ahead, not just for science, but for humanity as a whole. The promise of CRISPR and the infinite possibilities it holds are exciting, but they are also daunting. We stand at a crossroads, where the future is both within our grasp and yet unknown. As we continue to explore, to innovate, and to push the limits of what is possible, we must remember that we are not just changing the world around us, we are changing ourselves. The journey of science is intertwined with the journey of the human spirit, a journey that asks us to consider not just what we can do, but what we should do. Think, for a moment, of all the other questions that await us beyond CRISPR. What if we could harness the power of synthetic biology to create entirely new forms of life. What if we could use gene editing to not only cure disease, but to enhance human longevity, intelligence, or even creativity? What if we could engineer the perfect environment, where food, water, and energy are no longer scarce, but abundant for all? These are the questions that science will continue to explore, questions that will shape the future of life on Earth, and perhaps beyond. As we dream about these possibilities, we must also remain grounded in the reality that each step forward comes with challenges and responsibilities that we must face with wisdom, compassion, and foresight. The future of genetic engineering is not just a technological challenge. It is a philosophical and ethical one. As we continue to unlock the secrets of the human genome, as we push the boundaries of what is possible, we must ask ourselves, how do we navigate this journey responsibly? How do we ensure that the benefits of these discoveries are shared by all, that the power to alter life is wielded with care, and that we honor the diversity and complexity of the world around us? The answers to these questions will not be easy, but they are essential if we are to move forward in a way that respects the sanctity of life and the values that make us human. That as the stillness of the night deepens, you are left with a quiet sense of awe, not just at the advancements of science, but at the profound mystery of life itself. With every discovery, we come closer to understanding the intricate dance of nature, the delicate balance that allows life to thrive. Yet, as we look toward the future, we are reminded that the journey is never truly complete. There is always more to learn, more to discover, and more to explore. And in this endless journey of discovery, the possibilities of life are as infinite as the stars in the sky. And as you reflect on the vast potential of the future, you are gently led to wonder, how will we navigate this ever-expanding journey, ensuring that the advances of science continue to enrich, rather than diminish, the beauty and mystery of life itself?